Oh, hello everyone. We are very pleased to welcome you to this conference co-organized by the French Foundation for Biodiversity Research and the Center for the Synthesis and Analysis of Biodiversity in France. So this conference is dealing with the FISHGLOB project, which is dedicated to fish biodiversity facing global change. So it is currently a project under the umbrella of the United Nations of Ocean Decade Science. I'm Bastien Merigot. I am associate professor at the University of Montpellier in France in the Marine Biodiversity Exploitation and Conservation Lab. And I am the coordinators of FISHGLOB project with Aurore Moreau, who is a scientific consultant in France now, and also Deng Palomares, who is researcher at the University of British Columbia in Canada. So this conference is occurring in the frame of a workshop we had this week with our group in CESAB in Montpellier. And so before we start with scientific presentation, we would like to give the floor to the representative of the main founders of this workshop and conference, who allowed us to meet that time in Montpellier in person. So I would like to give the floor first to Philippe Combette, who is the Vice President of the Direction of Innovation and Partnership at the University of Montpellier.
Good afternoon, everyone. Ladies and gentlemen, I regret to inform you that Mr. President Auger is unable to join us today. However, he asked me to extend his warmest regards and to welcome you all on his behalf. For those who don't know me, I'm Philippe Combette, Vice President in charge of Partnership and Innovation at the University of Montpellier. It is my great pleasure to welcome you to the beautiful city of Montpellier. Let me take a moment to tell you a bit about the University of Montpellier. Our institution was founded back in 1220, and for the 8 centuries, it has been deeply rooted in this city and region. We are proud to host the oldest continuously operating medical faculty in Europe and the oldest botanical garden in France. These are just a couple of examples of the rich history and legacy we continue to build upon. Today, the university is a hub for education, research, and innovation. Together with research organizations, institutional partners, and our international network, we are advancing a scientific agenda that supports research excellence and the practical application of its results. With nearly 80 research laboratories and over 5,000 researchers, the work carried out at our university consistently garners international recognition. The university also coordinates a permanent e-seat initiative, Science, Innovation, Territories and Economy, an ambitious program that brings together 16 partners around 3 fundamental pillars. The first one, feed, developing innovating agriculture to ensure food security and environmental quality. Care, enhancing human health in the context of changing environments. And protect, driving a transition toward a more sustainable and environmentally friendly society. Given our location near the Mediterranean coast, in one of the world's largest wine production regions, and in areas grappling with water research challenges and population growth, we are well positioned to contribute meaningfully to global conversation about sustainability and innovation. That's why hosting the FISHGLOB International Conference is such an honor. This event is not only a valuable opportunity for scientific exchange, but also a significant contribution to the United Nations Decade of Ocean Science for Sustainable Development. Under the framework of the Ocean Decade, FISHGLOB is more than just a research initiative. It's a call to action, a global effort to address the profound and complex challenges facing fish biodiversity in a rapidly changing world. Oceans and their biodiversity are vital. They sustain ecosystems, support human health, and contribute to the well-being of communities around the globe. Yet, we know that marine ecosystems are increasingly at risk. Climate change, ocean acidification, habitat destruction, and overexploitation are reshaping the distribution, abundance, and survival of fish species worldwide. These changes are far-reaching implications for ecosystems, economies, and the millions of people who depend on fisheries for their livelihood and food security. FISHGLOB embodies the core principle of the Ocean Decade, fostering collaboration, integrating knowledge, and advancing the science we need for the ocean we want. Its mission is not only to deepen our understanding of fish biodiversity, but also to translate that knowledge into actionable strategy for sustainable management. Strategies that protect biodiversity, sustain livelihoods, and align with global commitments to sustainable development. This conference is a unique space, bringing together brilliant minds for around the world. It's a chance to share insights, discuss challenges, and most importantly, explore bold and collaborative solutions. Together, I'm confident that we can make meaningful progress in addressing the interconnected challenges for biodiversity conservation and global change. Thank you for being here. Special thanks to Aurore Moreau, Bastien Merigot, and Deng Palomares. I wish you all a successful and inspiring conference. Thank you.
Many thanks, Philippe, for your introduction, and once again, many thanks to your support to organize this workshop and this conference. Now we would like to give the floor to Hélène Soubelet, who is the General Director of the French Foundation of Biodiversity Research in France.
Thank you very much to be there. I'm very happy to be a part of this conference. I'm sure this will be extraordinary. Thank you also to be there despite the little change we had to make this last time. I would like to say a few words to present you the French Foundation for Biodiversity Research, so FRB. First of all, our main goal in FRB is to foster the dialogue between the scientific community and the society. And for that, we have been established by two main ministries in the perimeter of FRB, which are Ministries of Research and Ministries of Ecology in France, and by 8 public research institutions, which are now our founded members. So BRGM, CIRAD, CNRS, IFREMER, INRAE, IRD, et le Muséum d'Histoire Naturelle. And they were joined in 2014 by LVMH, which is our only private organization in the FRB board, and then INERIS in 2016, and then two other, last but not least, the University of Montpellier in 2017, and the French Biodiversity Office, OFB. So FRB, what is FRB? It's a body, and we try to do 3 main goals. So the first one is to better understanding the biodiversity, exploring its complexity, its challenges as well, the diverse relationship between the human activities and all forms of biodiversity and ecosystem, the variety of issues at stake, and to try to clarifying some controversies in order to support decision making. The second goal of FRB is to work together. That is to say, to co-develop and implement collaborative projects for and through biodiversity research to respond to societal demands at the national scale, but also at international level. And then the third goal is to talk about the future. So envisioning biodiversity scenarios, for example, to better plan for a more sustainable world and to identify some knowledge gaps. We have, for example, our own call. Every 2 years, for example, we launch a CESAB call with a few projects which are funded. We have also some joint calls. So one example is the call we launched with the Ministry of Ecology in France about infrastructure and their impact on biodiversity. Of course, we published some research results. For example, this one in October 2021 about lake temperature in the time of climate change, the result of one of our funded projects in CESAB. And we communicate about biodiversity, for example, through the CESABinar, so the CESAB webinars. We also are doing a lot of activities, some other activities such as evaluation of public policy, for example. I present you this evaluation we made on the global biodiversity framework before it was published within CBD, and we analyzed all the targets and try to know to show if the national strategy was aligning with this global biodiversity framework or not. And last but not least, we organize also some trainings. And for example, the new one in 2025 will be IA for ecologists, a toolkit especially for young researchers. So the CESAB is our main program within FRB. It's a dedicated space for the research and biodiversity synthesis. So we are gathering, the groups are gathering and pulling data. They are consolidating existing knowledge, developing advanced statistical methods to provide some assessments of biodiversity and model its future. So we develop major methodological advancement, for example, in handling heterogeneous data, creation of K-disciplinary databases, some example, tropical forest, soil invertebrates, intermittent river biomes, weed crops, and the functional diversity of trophic networks. We also organize a workshop, of course, and seminars, for example, on data management, as well as many training programs for researchers. And we organize also the networking within CESAB with other international synthesis center, including the Brazilian Symbiose and the Swedish Center in Europe, and of course the Canadian Center. So we generate new scientific principles and knowledge. We try to rapidly integrate science into policy making by dealing and talking with policymakers and other stakeholders. We catalyze collaboration across discipline, across private and public organization, and even in the countries level. And then we engage the diverse public, private, scientific, and non-scientific partners. And to finish, just a few numbers about CESAB. So in 2025, we will be allocated almost 2 million euros for the functioning of the center and the functioning of the scientific groups. We will have 35 active scientific groups in 2025, including 18 co-funded public or private partners. We will organize 6 training sessions, and of course we maintain diverse call formats. So that is to say, data synthesis project, knowledge synthesis research, or data share and ID share. These new forms of doing research within CESAB. And to finish, I would like to thank a lot the engagement and dynamism of our postdoctoral researchers, and of course the dedicated team of CESAB within FRB. And I wish you a very nice conference today, and I hope we will all learn a lot of things. Thank you very much.
Many thanks, Hélène, and many thanks to FRB also to support us since the beginning, for 4 years ago that we have started these projects with you. So many thanks also for your introduction to FRB. Now we are going to give you a recorded talk from Nicolas Mouquet, who is the scientific director of CESAB, who could not be able to join us in person today.
Hello, everyone. So first of all, I'd like to say how sorry I am not to be with you in person today. As you may know, there's currently a strike in France, and some of us are protesting against the government policies, and I'm on strike with them. Right now, I'm probably out on the streets demonstrating with others. But let's get back to this conference. Originally, we planned to welcome you in a beautiful amphitheater at the University of Montpellier. Unfortunately, we had to cancel and move this conference online. It's a disappointment because the CESAB team worked hard to organize this event, and I'd like to start by thanking them for their efforts, enthusiasm, and dedication. This enthusiasm reflects the special relationship we've built with the FISHGLOB group over the years. At CESAB, we strive to create a close supporting bond with the groups we host. We operate like a small family, offering flexibility and opportunities wherever we can. However, we still are a small center with limited resources, and sometimes we can't fully support the groups as much as we would like. But we do our best. And FISHGLOB is a perfect example of how we've managed to do a lot with very little. It all started in 2019 in a partnership with the Canadian National Synthesis Centre for Ecology and Evolution and the French Embassy in Vancouver. We launched a small joint call for proposals to fund just 2 groups without postdocs and with only 1 meeting in Canada and 1 in France. When I saw the FISHGLOB project led by Bastien Merigot and Maria Palomares, I was struck by their ambition and vision. It was clear that they were inviting us to join an incredible adventure they had already started, and I felt like a privilege for the CESAB to be part of it, rather than the opposite. And I was right. This group has been one of the most dynamic and responsive I've seen in 6 years at CESAB, and they did this while navigating the challenge of COVID. Thanks to their enthusiasm and the importance of their work, we were able, with the help of the Canadian Centre and the French Embassy, to fund an additional meeting in 22. Bastien, as well as Aurore Moreau, also visited the CESAB multiple times to collaborate with other groups. Bastien then kept the momentum of their group alive very well, publishing major scientific papers and achieving recognition as a United Nations Decade Action in 23, highlighting the significance of their work. This year, with support from the University of Montpellier and the FRB, as well as the Canadian Centre and the French Embassy, we managed to bring the group back for another week of meeting at CESAB and organize this conference for the scientific community. Overall, the FISHGLOB journey shows that even with modest funding, persistence can create a snowball effect and lead to great outcomes. And I am convinced that for FISHGLOB, this is just the beginning. The best is yet to come. I'm incredibly proud that the CESAB team was able to support them, even modestly, during this phase. It validates our philosophy to support groups, give them time to work in a calm and flexible environment, and help them secure additional funding beyond what we can offer. Bastien, then Aurore, it has been a true pleasure to support you over the past 4 years. I deeply thank you for returning here to share your work so generously with the scientific community. Thank you to all the members of your group for having been so active during these years and for coming back to Montpellier again. My thanks also go to all the participants for attending and, of course, to the CESAB team who make all of this magic possible. Thank you, everyone, and I wish you a great conference. I hope I'll be able to join you later today. Thank you.
So many thanks, Nicolas, for your kind words. I will say to you when we will meet again. So now we are We are going to start the scientific presentation related to FISHGLOB. I will provide few elements of context about what is FISHGLOB and the link with the United Nations Decade of Ocean. And it will give me also the opportunity to introduce my colleagues who are here today. And I'm very glad we are all together because they come from different countries far away, from Canada, from US, from Norway. And since the beginning, we co-created this project 4 years ago. So it's a real pleasure to have this conference all together to provide you the different activity we had until now. So first of all, I would like again to thank you very much to the FRB and CESAB for the support since the beginning of the project. All the CESAB staff also who organized this conference. Basically, it will have been in person at the University of Montpellier, and so they spend a lot of effort and time to create this event. Hopefully, we could shift it remotely, but I would like to thank them to all the effort they made until now. I would like also to thank the Canadian Institute of Ecology and Evolution, who co-founded this workshop and conferences, as well as the University of Montpellier and the French Embassy in Canada. So the context of our activity is linked to the global change and the different impact on marine biodiversity, especially on fish biodiversity. And recently, it has been recognized that around 66% of the ocean show an increased human impact, mainly driven by climate change pressure. So it is well known that surface temperature can have an effect on marine community, but there have been also evidence that there is some bottom marine heat waves that occur in the ocean. For instance, here you can see the mean intensity of bottom marine heat wave on continental shelves across the North American continent. So you can see that the average intensity can reach until plus 5 degrees. And what is interesting is that this kind of bottom marine wave can occur at the same times of surface marine heat wave, but sometimes it's not the case, and they can last longer and more and can be more intense than surface marine heat wave. But we don't know yet precisely the effect that this kind of bottom heat wave have on marine biodiversity. So we will see later in the presentation of Alessia Freston and Manin Pisky how this effect can impact fish biodiversity. There have already been some evidence of impacts of temperature rise at local scale, regional scale. This is a work from James Thorson, who is also part of our group, who shown that for 4 main species assemblages, there was a shift northward for some species of commercial interest associated with change in bottom sea temperature and loss of sea ice, but also that the responses varied according to the species. Some of them could maintain a high density in the thousand habitats. But more generally, if we consider a predicted impact of sea temperature rising, it has been shown that there is a different degree of impact according to the zones across the world, and that the species turnover can reach a huge amount of percent. For instance, in the tropical area, almost 40% are expected to change in terms of composition. So in this map, you can see the rate of change for new species and rate of change of loss of species. And so you can see the spatial heterogeneity according to the zone we consider in terms of prediction. And this kind of species composition shift can also affect fisheries in terms of catch proportion and stocks. Here you can see a predicted map of this change performed by Giuliano Palacios Abantes, who is also part of our group and who will present with Nancy Shackle later this topic. And they notably predicts that almost 23% of the stock will move during the next decades, and that almost 80% of the exclusive economic zone have at least one stock that will move, and that could also create some potential conflicts for resources across the nation. One also motivation to study fish is its role in ecosystem functioning and services. Now we know that fish have an important role in carbon sequestration, and it has been shown that fisheries extract a lot of this potential carbon sequestration by ocean. And we also know that fish is an important source of protein and micronutrients across the globe. So the thing is that global change affects in a different manner the globe in space and time. And we can see on this map the spatial heterogeneity of sea surface temperature and also of fishing efforts. So if we want to have an exhaustive assessment of the effect of global change at the scale of the globe, we need to have dedicated observation program to monitor this change and biodiversity shifts. So one possible observation data that can be used are those from scientific bottom survey, because some of them have a long time series that can last at least for 10 years. They can allow us to assess individual counts and biomass, not only presence and absence of species. So we can go more into precision about the impact of global change. But they have a different sampling strategy, temporal and spatial extent, and also different taxonomic scope. And they have also a various statute of accessibility. Some are public accessible and some other are private accessible. So the main aims of the FISHGLOB project was first to create a network of data providers and experts on scientific bottom survey across the world. Then to create an inventory of the existing surveys and to provide to the scientific community the metadata of public survey as well as full data, meaning abundance of biomass data, for instance. And this in a homogenized format after specific processing. And also we aim at providing contacts of data provider that own the private data. And based on this data set, we aim to address key resource questions related to global change impact on fish population and biodiversity. Overall, to finally create an infrastructure to support science and biodiversity conservation and fishery management. So currently, what is FISHGLOB? So FISHGLOB, in fact, is crossing different key aspects. So the first one is a network of data provider and expert on scientific bottom troll survey. Currently, it gathers more than 100 people, and we are very grateful to this data provider and expert for the work we have done together this last year. It was essential they could participate and exchange with us very closely. We create through this network a database which encompasses both metadata and full data set. Some of them are a very long time series of survey, as you can see on this slide. And FISHGLOB also consists in a synthesis group, so funded by the previous sponsor we represented, and also by a steering committee formed by 11 people who are all here together today. So just to tell you quickly about the genesis of FISHGLOB. So it started 5 years ago during a side meeting of an ICS conference from the initiative of young scientists, so Aurore Moreau, Lorraine Pécuchet, and Romain Frela, who discussed with Nancy Schachtel and James Thornton to the research activity to make an inventory of existing bottom troll survey data set, and especially to create a database with metadata to know exactly where and when scientific bottom troll survey occurs. Then starting to 2020, we create a synthesis group from a joint synergy call from the France and Canada to create a full data set with abundance and biomass data from these surveys and to starting at addressing some key research questions. And starting last year, we were under the umbrella, we are under the umbrella of the United Nations Decade of Ocean Science, and I will speak about more just after this. So about the network and data. So here you can see the map of the network of experts and surveys we are in link with, and also a full data set so that it embed biological data of surveys on the American and European continent. So these are linked to two papers you can find thoroughly on the web. And our synthesis group is formed of a researcher and teacher researcher from Norway, Canada, US, France, and also Netherlands at the very beginning. So I will let everyone introduce himself all along the presentation. But as you can see, we are a group for different institutes around the world. And what about the link with the United Nations Decade of Ocean Science? So first of all, to introduce this initiative, it seeks to stimulate ocean science and knowledge to reverse the decline of the ocean system we currently face and to create new opportunities for sustainable development. It aims at increase the interaction between scientific community, policymakers, and also civil society around an agenda of action during 10 years. And currently, this initiative gathers more than 50 programs, and these programs embed some projects. And FISHGLOB is currently one of these projects. So we are almost 40, 60 projects. And if you are more interested in the structure of the ocean decades, you can see here the three main objectives of the decade. So to identify required knowledge, generate comprehensive knowledge, and increase the use of ocean knowledge, and to help to address this objective. So you have different actions, so program and project in link to 10 decade challenge. So one of the program existing now is Supreme, who is more interesting in marine ecosystem dynamics and to provide decision tools, and is more interesting in the forecast, prediction, and projection of a marine ecosystem. And FISHGLOB is under the umbrella of this program. And now we are on the way to create an infrastructure and a platform to gather both scientists, government agencies, and stakeholders in order to facilitate interaction and establish baseline to understand and predict the fish biodiversity change and to help identify management action. So if you have any interest in FISHGLOB, if you want more information, or if you want to contribute to our project, please go to our website where you could find a form that we will receive. And you have also here provided the email to join us if you are interested in. So now I will give the floor to my colleagues who will present all along the few next hours some complementary topic we had in FISHGLOB. So we will go from the data set, the infrastructure, to new methods to better assess the change in fish community and beyond. And then we will have some presentation related to more ecological aspects related to the dynamic of fish population and community until the applied aspect of guidance for fishery management. So thank you for your attention. We can have some question maybe in the chat that we will respond during the next presentation because we are already a bit late. We will go ahead with the presentation. Thank you with the presentation of Aurore Moreau, who's a scientific consultant now in France and with the COPIA and who is very much involved since the beginning in the project as one of the main coordinators.
Hi, everyone. Thank you for joining this conference. So I'm going to give around a 50-minute presentation on the FISHGLOB dataset, international community building and infrastructure. And I will take it back a bit from the beginning and how we started building this project and dataset. All right, so we are, as Bastien introduced, working on bottom trawl survey monitoring around the world. So there are many ways that we sample marine communities in the ocean. So different ways are acoustic dredges, traps, trawls, many different types that are represented on the left here. And bottom trawl surveys are actually trawls that we put towards the bottom of the ocean with boats. And we physically sample communities that are living on continental shelves and slopes. So these communities are mostly fish and invertebrates that live close to the seafloor and give snapshots of the community in space and time from 0 to 200 meters deep and even deeper than that. So these are snapshots of how this monitoring actually occurs in different waters of the ocean. So here I'm showing photos of the Western Black Sea, Mediterranean Sea, and Iceland. And these photos were provided by some of our colleagues that we've been reaching out to in the last few years, since 2019. So why we've been thinking about focusing on these surveys is because they're very numerous and very data-rich sources of information on marine biodiversity. But they are conducted in different regions of the world with no real cooperation and coordination across regions, while they can give very important information about biodiversity change. And so when we started, we were wondering what data exists around the world from bottom trawl surveys, who might be interested in scientific cooperation and why, what can be achieved with international collaborations on this survey data, and how can cooperation help conservation and management issues. So we'll try to answer some of these questions, and the following presentations will keep showcasing how we can use this data. So in our efforts, we've first focused on survey metadata. So metadata is just giving information about what kind of data exists, such as survey metadata focusing on what is the name of the survey, what institutions are carrying the survey, what is the gear that is used to sample marine communities, what is the contact information, and who are the experts. And the event metadata gives information on the actual samples, the time, the date, the effort, the location, and sometimes the species that are sampled. And so all of this information gives information about what data are out there without having to actually get the data. And I think focusing on metadata allowed us to search for data in an easier way and really try to reach out and get information. So in our efforts to create a metadata inventory since 2019, we've used our contacts and we've reached out to new people that we found, searched fisheries institutes, and looked in the literature to be able to reach out to as many people as possible working on bottom trawl survey monitoring. And we are very grateful that so many people answered us and that this has been an ongoing effort and that we've seen so much interest in working with us in collaborations. And so in this effort, we've really created a network of experts that we are hoping to continue. So this metadata inventory led to the discovery of 95 bottom trawl surveys that covered the last 20 years since 2000. So 95 bottom trawl surveys is pretty huge, and each of these surveys are regional and they cover different spatio-temporal areas. They represent more than 280,000 samples, and we also assessed the availability and the visibility of these surveys. And we found that 59% of the metadata that we found in volume is actually not publicly available for many different reasons. And most of the publicly available data come from Europe and North America, which is a pretty classic pattern in the history of data availability and comes from the science and the fact that open science has developed more in rich countries. So it's also very important to notice that there are some surveys that have like a mixed level of availability or that can be accessible through requests directly with the data experts. So having collaborations with all of these experts around the world is really interesting, just to exchange information and also to know what kind of information exists around the world. So in our project, we've not only focused on metadata and building relationships, and we've also worked on gathering and integrating the available data that was open. So the data that is open really is getting at the sample information about which species are sampled, what is their occurrence, what are their number, and what are their weight, which has been the most information that we are covering across the different regions, although more information from these surveys actually exist. So in our synthesis group, we've actually created a database of these publicly available surveys that was published earlier this year. So it covers 29 public surveys that have been integrated. So all of the spatial footprint is displayed on the map here, and it mostly covers European and North American waters. So it's actually covering 18 countries' national waters. And these surveys, these surveys are publicly available, so everybody can actually access it. So these surveys are also, although they are very similar in their principle, they are very different in their design and in their spatial temporal coverage. So here I'm showing in the row all of the survey name for the public surveys, and the colors show the sampling intensity through time. And so we have surveys that started in the 1963 until 2021. And some surveys are actually much more recent or don't happen every year and sample different taxa. So we cover now more than 2,000 fish taxa in this data set, and we have more than 200,000 samples. So the samples in the trawl surveys are called holds as well. So to be able to process these surveys, because they are disparate and not necessarily coordinated and available in different formats, we had to develop a processing workflow that can be applied to any additional survey. So the first step is to do taxonomic homogenization steps using broad and updated databases of taxonomy through validation and harmonizing steps, as well as thinking about data harmonization with selecting the good quality data and arriving to survey holes and taxon standardized units. In the second step, we've tried to verify the quality and the spatial temporal coverage of the data. So the first one was really about thinking of trying to identify weird species that may be occurring, because sometimes they need quality checks with the survey experts, or try to think about what is the spatial temporal coverage of the data. So this is an example of France and of the Bay of Biscay, where we've ordinated years and grid cells in space, where we try to see what is the holes that you need to select to have a good spatial temporal coverage, and that can allow spatial temporal type of studies. So on the right map, all of the holes that are flagged in blue and in green show the holes that we would need to remove to have, that we would need to keep, to have like this good spatial temporal coverage. Finally, we've also done a few technical validation steps, both at the survey level and cross surveys. So these summaries really get at checking the validity of the data, both among our team and with the consortium, or any user can actually access this and check what is the data that is available. So that covers the format of the survey data, the sampling intensity, sampling variables, biological variables, extreme values, as well as temporal trends and spatial patterns. And finally, we've developed a few filters in this dataset that can allow users to easily check and choose which taxonomy level they're interested in, which standardized unit they're interested in, and try to filter as well sampling depending on the survey, on the season, and so on. So this data set is completely open. It's available for everyone through the GitHub repository. All of the code is public. All of the processing is public. Anyone can use it. Anyone can submit comments or concerns that they found. And they can obviously download and use the data set themselves, either on the GitHub repository or on our website that Bastien already presented earlier. And you can use the code here to access it directly. So I wanted to go back a little bit more on the community that we've created while starting with datasets. So I think one of the very important things in our work has been to create communities. So I would say at this point, we have a community of regional data experts and a community of data users. Sometimes a person can be at the same time a data expert and a user, but not necessarily. And having a technical infrastructure and a data set has really allowed connecting these communities together and try to think as a group how we are processing and using bottom trawl surveys. So in this process, it's really important to have values and principles that we developed as a community to try to see how we can actually maintain partnerships that are meaningful in time. Such values are the fact that we're interested in open data and open science as guiding principles. However, we are also respecting data sovereignty and the fact that some surveys are not available and that it's not necessarily our role to advocate for changing data availability. But however, what we're trying to do is to maximize partnerships and collaborations. Whatever is happening, we're just really happy to collaborate with people who have expertise on demersal communities and surveys. We are also interested in capacity building and grow an international community. And finally, giving credit and visibility to our consortium members. So talking about credit and visibility, so I listed here all of the persons who have been active members of our consortium and our data set. And I'm very hopeful that there's going to be many more in the future and very excited that so many people have contributed to our project over the last few years. So I just wanted to give a few words to the fact that while exchanging with so many diverse experts, I've heard so many stories about bottom trawl survey monitoring, both about global change in surveyed regions, as well as maintaining monitoring, and the fact that maintaining bottom trawl survey monitoring is actually a real challenge nowadays, while it's a really important type of sampling that really allows knowing what is happening in the ocean. The fact that the reputation of bottom trawling in society has affected a little bit the maintenance of bottom trawl survey monitoring in the ocean and the funding. And the fact that there is a lot of competition for ocean space and that, for instance, the development of offshore wind farm may affect the continuity of good sampling in space and time. There's also been intersections between geopolitics and science in our project, which I think has been very interesting to know and to try to know more about, and as well as marine territory sovereignty. So I'm very grateful to have heard about this, and I'm hoping that our team will be maybe better at communicating all of these stories moving forward. So I will end here by saying that we have created a new international scientific consortium of experts over the last few years, that we've put together a new data set that is available for the community. We've integrated expertise from survey regions up to global data process products, and that we are a platform for knowledge exchange, and that our project also has ocean literacy opportunities, which I think are very important, especially in the context of the ocean decade. And I'm going to wrap up here. So thank you, and please let us know if you have any questions.
Many thanks, Aurore. So does anyone have any questions? Please raise your hand or let us know by the chat, and we will give you the floor. We have time for questions, so don't feel free to ask. We welcome Deng and Rob for the next presentation. We are going to talk about the future of the FISHGLOB infrastructure. So we are working very actively to build this infrastructure. By also responding to call for that. It's not easy to find money for find the infrastructure. So currently, Rob Guralnik and also Malin Pinsky are working out for that on the US side. And we are making also some tentative in Europe. With Denk Panoramas, we will try also to an European call soon. So we will see. But for about the content, Deng and Rob will present you more precisely what we are looking for in terms of infrastructure.
But before I begin, there is a question in chat for Aurora.
Okay.
Yeah, do you want to come back and answer it?
What is the question?
Sorry, taking the spot again. Okay, so complementarity with other types of data sets and sampling methods. I would say not yet. Not really, because we've been so busy with just one monitoring type. We've thought about complementing the bottom trawl surveys with the pelagic trawl surveys, for instance, which are relatively similar, but we've not done that. So there's been thoughts as well about combining bottom trawl surveys with video surveys that maybe then she will talk about it later. So thank you for the question.
Okay, so we can take care of the additional question, yeah.
We are always waiting for new data sets, always ready to integrate new data sets. Yeah. We are also very interested in maintaining contacts of who has new data sets. And we don't always have capacity to integrate them right away, but yeah. And also everyone can do it if they want to, because the code is available and we just need to coordinate with us, basically. Acoustic surveys. We haven't really thought about using acoustic surveys, but we would love it if a new group was doing acoustic surveys and wanted to work with us.
Yeah, because it is a huge effort to gather, standardize, etc. The data. So it's also the reason why we focus on bottom trawl surveys. But hopefully, if there is some people who are interested in considering additional kind of method of survey and stuff, you are very, very welcome. And because the procedure and stuff are set, we can easily share our experience with that, and we can welcome any additional initiative in that sense.
And also, like I see a question about the fact that we have very large spatial temporal scales, and that's true. But I think in the aim of understanding global change on marine communities, it doesn't really matter if we want to do cross-monitoring studies. Then it's interesting to have any kind of monitoring, even though it's not the same spatial temporal scales. And I think also as a community, from a broader perspective, it's really important to have similar standards of how we process data. So that also will be covered in the next presentation. So maybe that's a good bridge for the next talk. Right.
Okay, great. Thank you very much. So now we are going to the next topic about the future of the FISHGLOB infrastructure by Denk Panoramas and Rob Guralnik. So please.
Great, thanks, Bastien. Thank you so much, everyone, for your attention, for sticking around. My name is Rob Guralnik, and I'm sitting next to Deng Palomares, and we are co-presenting today on FISHGLOB infrastructure, past, present, and future. And although we're going to focus on FISHGLOB, we also want to expand out and discuss how FISHGLOB connects to other allied data and knowledge infrastructures. So I'm going to start us off and cover one larger picture issue before I turn it over to Deng. And then I'm going to pop back on towards the middle of the presentation to cover a little more about interoperability. So I'm going to start by coming back to the slide that Aurora showed that I think is really important. It shows this nested set of data and metadata generated from any scientific bottom trawl survey. And this nestedness applies not only to scientific bottom trawl surveys, but whether we're talking about that or whether we're talking about eDNA or passive acoustic surveys, this really applies broadly. And while Aurora discussed this briefly, you may have wondered, well, what exactly are the metadata standards that can be used to help make FISHGLOB data interoperable? And how do we even capture the often complex metadata that are needed to represent surveys and inventories? So we're going to come back to that question and kind of roll that around for the next few minutes here. And what I'm going to do now is finally turn this over to Dang, who really is an expert in kind of thinking through the logic of connectivity between infrastructures and building out these large-scale infrastructures for scientific research. So over to you, Dang.
Do we need to change? Maybe let's just like that. Oh, yeah. All right. So this is just a short recall of what the fish-based infrastructure is. Now we know from Aurora's presentation that we have a survey format, and that is being homogenized across surveys. There is cleaning of the taxonomy so that everything is on the same page. And while this is happening, experts are able to do feedback on the processing. And after that, there's taxonomic flagging in time series, spatial temporal standardization, and more importantly, the collection of traits, trait imputation, and link to surveys. Now, how does all look in an overview of the system and how FISHGLOB is situated around the digital world of data sets and information systems? So here we put FISHGLOB in the center of things, and we have our arrows back and forth or towards FISHGLOB or going out of FISHGLOB. So these are the different components of this infrastructure, and we will then go into each one of them. So Aurora gave a really good presentation of how data and data contributors are connecting with FISHGLOB. So that is part of this formatting and homogenization of the surveys and the feedback from experts. And all of this is then put into the use of publics, but the public's use freely via the internet. Then you have the spatial temporal standardization that feeds from FISHGLOB to biogeographic information systems. And for example, OBIS and Ocean Adapt, both are getting information from FISHGLOB. Now, the taxonomic flagging in time series is when you have a database or a biogeographic information system that actually uses the time series format or has a temporal infrastructure, because FISHGLOB has both. Now, this is how we get the taxonomy cleaned. FISHGLOB actually goes to or uses worms, and which is on the worms has fish-based taxonomy integrated into it, because FishBase is the fish expert for global species database of the World Register of Marine Species. And there is a tripartite memorandum of understanding between worms, fish-based, and sea life base, because worms is the taxonomic backbone of sea life base. Then fish-based and sea life base, that is, have traits data in it, and it's part of the information system. And that information is very important for FISHGLOB, because they use it for the estimation of biomass and other things. However, not all of the species in FISHGLOB would have the traits that they need. And then in comes or enters fish life, which provides the imputation mechanism so that traits that are absent from the fish base in data can be imputed and then used by FISHGLOB. So how does FISHGLOB relate to the wider user community? Now, our plan is to make FISHGLOB available via the European Ocean Science Cloud services, and we would need active data exchanges with other projects and other data aggregators. Already, the FISHGLOB is being used by the researchers who are associated with it right now, and you will see that later in the later talks, how FISHGLOB data has been used in innovative research ideas and synergies between different organizations. Now, I turn this back to Rob, which is who's right there.
It's musical chairs here at the meeting. So what I want to cover to kind of wind us towards the finish line here is talking about one last piece of this puzzle around interoperability between FISHGLOB and other data aggregators. And in particular, I want to focus on the ability to make these species occurrence data and the metadata about inventories and surveys broadly available and reusable and shareable in the best possible way according to FAIR principles. And the core data loop that we're going to focus on here is where FISHGLOB connects to OBIS and to the Global Biodiversity Information Facility. If you don't know, OBIS is the Ocean Biogeographic Information System. It's a really important infrastructure for serving up the primary occurrence data and a lot of the metadata and a lot of the information about surveys in a standardized format. GBIF is sort of an overarching project to connect all of these data resources together around species occurrences and biodiversity data. It's an international effort. It's been very successful. And of course, we have data contributors who are providing us data. We're helping us harmonize, standardize, and make interoperable these data. And we're hopefully going to be able to make sure that these data interoperate well with these larger aggregators. So the challenge is that unlike GBIF and OBIS data, and this is true in some ways, and some OBIS data are very well represented around survey and inventory metadata, but for many resources on GBIF and OBIS, we're not just talking about simple species occurrences. We're talking about the complicated inventory metadata that Aurora mentioned and Deng and I have mentioned as well. And really the goal here is to build linked open data frameworks that use shared semantics and globally unique identifiers to help link that all together into a bigger system. Okay, so let's talk about the pieces of this just really briefly. I won't dwell on this, but I'll just cover it in the large picture, in the simplified larger picture. But core to this is using standards that have been developed by standards bodies. I don't know how many of you in the audience know what Darwin Core is. It's a standard that is ratified and has been well used for representing species occurrence data. It represents the simple stuff about what is this taxon? Where was it collected? Who collected it? When was it collected? And this kind of species occurrence data and metadata is primary information we need about occurrences and specimens. Okay, but like I mentioned before, just having that information is not sufficient when we have to deal with the inventory and survey metadata and the event metadata. One thing I think is missed about Darwin Core is that it's actually representable both as an occurrence core and what's called an event core. The occurrence core is where you have an occurrence identifier linked to an occurrence record, and then there's information about the occurrence record like a measurement or a factual statement. And you could also think about that occurrence core linking to like a process that generates a DNA sequence linked to that specimen or occurrence through a material sample ID. The event core is like turning Darwin Core on its head. You think about it as instead of occurrences, events, there's an event ID. That event has metadata like where was this collection happening? When did it happen? How long did it happen? Those generate their own extended measurement or fact sets of information, and occurrences are linked to that event ID and to the event core directly. So you have a single event that generates multiple occurrences and you can map that, right? So that's what Darwin Core does. But even with the event core, we still can't really make the interoperability work. And the missing piece is really about that metadata about surveys and inventories. And so I've been involved for many years in developing an extension to the Darwin core. This all works in the same publishing system that has been developed over many years that allows data to be interoperable at the data level, at the publication level. And that extension is called the Humboldt extension. The Humboldt extension really basically allows us to represent all that rich information about inventories and surveys. There are 55 total terms that are shown in colors around this event core. We have geospatial scope and habitat terms. We have temporal scope terms, taxonomic scope terms, organismal scope terms, methodological descriptions, and sampling effort terms. And they're all shown in the right-hand side of this figure, where you can see all of those specific terms. And then on the left-hand side, you can kind of see how it's organized. What's really powerful about this is now we have a mechanism to represent all the rich metadata we have and are going to be representing in FISHGLOB in a way that's built interoperable with GBIF and OBIS, right? So that's a powerful way to begin to do this. And even though we have all this, one could argue we're still not maybe 100% purely going to be able to use just these terms for all of the kind of complex ways we think about inventory and survey information from scientific bottom trawl surveys. But we're going to do 99% of the work with this set of standards. One last quick thing before I wrap up here. I want to be on time, make sure we're not going to go over here, but I want to talk about one last piece of this puzzle. It's away from standards and more towards how we implement the kind of working model for how FISHGLOB coordinates data around our team. And that has to do with using GitHub for data and issue management. And I won't belabor this. I'll just say that GitHub is a great, simple tool for us to use for basically having the power of, the power of the resources that GitHub has for basically allowing us to do git push, pull, merge, and compare requests. Data moves into FISHGLOB, and the workflows are all kind of managed in GitHub. And we can use issue trackers to help us kind of deal with the data management workflows that we want to adjust and improve. GitHub is great. I love GitHub. But one thing we also want is to be able to leverage the power of relational databases to help us do more. And so I'm going to just mention one last quick thing here. There is something called DOLT. And DOLT is like the child that was produced when you merge MySQL and GitHub. It's a beautiful thing because it allows us all the power of SQL for querying, for producing APIs, for being able to like build a better model for these data, all the complexity, while having all the power of being able to like deal with GitHub push, pull, merge, compares, all things we normally do, we can do beautifully in DOLT and DOLT Hub. So that's where we're going to go. We're going to build out this really beautiful system for doing this in a community way using these tools. So to finish up, I want to just mention that I've covered the cyber infrastructure layer in green, where we're talking about data and metadata standards and the database. There's this community process that has to happen with co-design workshops, community strengthening workshops, community expansion, that all fits in and kind of coordinates that social part that I think Aurora is so clear about making sure we understand it. It's not just about technology, it's building out this community. And in between here, we're going to be developing some really cool and interesting secondary products and value-add products that will help us kind of engage the community. So thank you so much for your attention. I really appreciate it. I'm happy to take any questions, and Dang as well. Dang.
Well, many thanks, Deng and Rob. So if you have any question, please.
There's one question.
Okay.
Do you trust the different providers at identical level? Are there any methods?
Come on over. You're part of our team anyway.
Yes, we do. That would then be fair to not trust everyone to the same level, I guess. However, we also have a way to integrate taxonomic flags that are not consistent in time if we have a taxa that has appeared, and then we have a way to go back to the providers and ask them what these are. And if I can add an anecdote here, we had somebody in their survey as a joke who had included the rooster because they were eating that on the boat when they were sampling. So we found it up. So I think our methods are validated in that process.
Yeah, great answer. I'll just say I think it is important to think about flagging data and making sure that we have flags and quality checks. I think that is a sufficient process for us. I think more than that, we get into issues around that are complicated around interpretation, right? So we do flag, but we don't do much more.
So an additional question?
We would let the present a goose.
Okay.
Yeah, please look us up. We'll do that.
Yeah, thanks, June, for your comment. Is there any additional questions or comments? If you feel that it's too long to write in the chat, please turn on your microphone and speak directly also, because sometimes it's long to write, so feel free to directly speak. We have a few minutes, so we can welcome any question or comments. We wait a bit in order if somebody want to.
We were so clear.
To write or
Bastien.
Yeah,
we were so clear. There are no questions.
So now we are starting presentations about the use of the FISHGLOB data to address research key questions, but also presentation dealing with new methods to deepen the assessment of fish population and community change, but also methods that can be applied to any other kind of species group. So these two next presentation will be done by James Tothons, who will present you first a series of methods and questions related.
Great.
So James, if you want to take the floor.
Yeah, thank you, Bastien. I'm Jim Thorson. I'll just try to get up here just from profile. And then I'm a statistical ecologist in Seattle at the Alaska Fisheries Science Center. It's one of the 6 science centers of the National Marine Fisheries Service. I'm really excited to be here. I would have been excited to be on strike as well, but you don't get to choose these things. Well, I don't at least. But yeah, and I'm here today to talk about several collaborations I've done with people here over the last several years. So FISHGLOB is a remarkable data set in many ways. One way to think about it is that it has this amazing spatial coverage. It's sort of a global data set. It has a unique temporal coverage for global data sets, so 40 years of data. And it's also this huge multi-species data set. So there's a tremendous amount of phylogenetic and taxonomic variation. And so, we have a lot of domain knowledge about habitat, temporal, and spatial information that we wanted to figure out how to bring to bear in our analysis of this unique multi-species spatiotemporal data set. Let's see, maybe I have to, there we go. So, this is obviously a cartoon here, but I would say a lot of science has, a lot of ecology uses things like GAMs or regression models. These are often useful for exploratory regression and are great for interpolating within, between samples or making predictions within sort of the same process that gave rise to your data. As we know as ecologists, you can also use regressions to make causal inference. You can infer what causes what if you have experimental data. But in this kind of global change context, we actually need to think about how do you make inference about structural relationships and causality using data sets that you collect that are not experimental. And so, the world of causal statistics has really been investing time thinking about structural models that can be used to extrapolate beyond samples that predict under changing conditions. And a lot of causal statistics, I mean, it's too much to summarize here, but one way to do that is to think about making structural models like directed acyclic graphs or representing a priori some assumed relationships among variables, and then conditional on that assumed relationship, you can make causal inference. And so that's sort of what we're thinking about here. So one study is this was published in Methods of Ecology Evolution. It was with Alex Andrews and at the Alaska Fisheries Science Center with me, Tim Essington at UW, and Scott Large at the Northeast Fisheries Science Center, published earlier this year. And I like to start with this because it's a paper where we actually went through and thought about how other fields think about structural modeling. So, economists often use something similar that they sometimes call simultaneous equation models. One way to represent a structural model is with this sort of arrow notation that we're extending to an arrow and lag notation here. And so this is a kind of, famous toy example of a macroeconomic model for, how economies function. The developer of this model, got a Nobel Prize back in the day for it and used it to model the Great Depression. But it's essentially this simultaneous equation where we think about how corporate profits and government spending and private spending affect investment and wages and inflation rates. And so, economists are used to thinking about these, economists, when they make a decision about government spending and how that might affect inflation, they have to be thinking causally. They have to be using a structural model. And that's what us ecologists should be doing too. And so, in this case, we can fit that model using this R package. And we get, again, what looks, this directed acyclic graph where, for instance, as capital stocks get larger, the model predicts that investment will decrease. As investments increased, you would expect private wages to increase. And as the private wages increase, you would expect consumption to increase. And so you can start mapping out these indirect effects via this chain of linkages in the, with their estimated magnitudes. This is often done with structural equation models in ecology. Part of what was new in this paper was that we started putting in time lags. And so this model has a time lag where government expenses affect private wages this year, but they also have a lagged effect on private wages next year. And so, ecologists also get it lags in species interactions, so it's applicable. Alternatively, fishery scientists often think about things like, direct climate impacts on metabolism, but also climate might impact forage availability. We might have missing information about some surveys aren't in every year. And so we wanted to think about how we do this. And we use this Eastern Bering Sea pollock, one of the, it's a $1 billion fishery in the Eastern Bering Sea. And so in this example, we, with the graphics department, we estimated effects based on multiple surveys in the Eastern Bering Sea. But, we conclude that increase in sea ice drives an increase in this cold near bottom water called the cold pool. As the cold pool increases, there's way more copepods, and there's also an effect on krill or euphausid density. When copepods or krill are higher in abundance, they also show up more in the diet of pollock. And when those two zooplankton species show up in diets, it contributes to greater overwinter survival of juvenile pollock and greater subsequent recruitment. So this is a way of thinking about climate to recruitment, which is a core problem in fisheries, but kind of breaking it down into different pieces that you can experimentally and observationally measure. So that's a time series example. We also, in the working group, wanted to think about the phylogenetic, this amazing phylogenetic diversity that shows up in FISHGLOB. And so thanks to the many authors here, I think everybody's a part of the group except Sarah Friedman at the Alaska Center, Jenny Bigman at the Alaska Center now, and others. I'm sorry that the ... cut it off. Yeah, so Aurora made this wonderful figure summarizing how we could think about this on a tree or a phylogeny. So at the top, we might have a phylogeny where there's some missing values. The phylogeny is shown as this tree. We might also specify a graph where we've got 4 traits. Trait 1 causes trait 2, trait 2 affects 3 and 4, and 3 also affects 4. We can represent that with this arrow notation, and that constructs a path matrix. And that path matrix and the tree are then assembled to make a big covariance that's used to impute traits in a generalized linear mixed model. So that's how this plays out in a phylogeny. In the end, you end up, we applied this to, 32,000, 32,000 described fishes and this database of traits, and we get these estimated effects where temperature affects asymptotic, an increase in temperature decreases asymptotic size, and that has a direct negative effect on growth rate, but there's also an effect of temperature on growth rate and so on. There's a bunch of interesting stories here, but one is that an increase in asymptotic length decreases the probability of a garter, of a guarding reproductive habit. So Ken Anderson has this story about how fish want to target a juvenile size that's a fixed ratio of the adult size, but that eggs can't be shrunk smaller than the size of a retina at birth. That retina is about 1 millimeter. And so as fish shrink down, they can't produce as many individual offspring as they want. And so instead they invest energy in protecting them, either by protecting the eggs behaviorally or by, brooding them in their gills. And so that sort of garter strategy is more prevalent with small fish. And that's what we see when we do this sort of cross-species comparison. We can also use those prediction, we can use that model to predict traits. And then thanks to Loren, we were able to classify them based on these 3 archetypes between periodic, opportunistic, and equilibrium life history types. And so I like showing this for fisheries people because I, as a fisheries person, I thought I knew what these meant, but I'd only ever really looked at periodic and equilibrium, most of the commercial fish over there. Opportunistic includes fish that are much more like insects, these 30-day long lived gobies that, are not typically commercially exploited. And so it's great to challenge your assumptions about what you know by looking across all of the described species. And then finally, there's another paper thinking about how to do this spatially. So with Sean Anderson at DFO, Pamela Goddard is a contractor at the Alaska Center, and Chris Rupert is also at DFO. This paper is an archive and still in review. In it, I'm just checking the time. I'm doing fine for time. Yeah. I should just ramble more. We've got sponges and corals that we study at NOAA, and it's a concern that bottom trawling might impact them. We've got bottom trawling where we can't do it in untrawlable habitats, rocky habitats where the gear might get damaged or we might damage the substrate. And then we've got people like here showing Kresimir Rupert that have made these sort of Arduino, these kind of cheap underwater vehicles that we can tow behind a vessel, and the images, using like a GoPro can be automatically annotated using AI neural networks. And so that's growing importance in the field of fisheries, these camera trap surveys. And so here we were interested in thinking kind of structurally about how we could combine something like FISHGLOB, where we have extensive bottom trawls, with these targeted campaigns to do video trap surveys. So this is a busy figure, and I'll walk through it slowly because I've got time. But so here we've got sponges and coral densities shown in S and C, and we've got rockfish and flatfish densities in the drop camera survey. So we've got 4 variables in the drop camera. And then we've got rockfish and flatfish and a bottom trawl survey occurring in the same areas. So here I'm looking at data across the Bering Sea and Aleutian Islands, which is two of the 30 regions that we have public data, 2 of the 100 regions that we have metadata for. And so The classic way that we do this is we'll use the drop camera to estimate coral and sponge density, and we'll use that as a covariate. We'll assume that we know that perfectly, and we'll plug it in as a predictor for densities of fish. And in some sense, that's well, let's see. So on the one hand, even simpler, you might think that there's no link between the drop camera and the bottom trawl, that they're measuring essentially different things. And so you might model how sponges and corals affect rockfish and flatfish in the drop camera, and that the bottom trawl is just doing its own thing. You might alternatively think of the sponges and corals as a predictor of the bottom trawl observations in that same spot, and that's what I'm calling a missing covariate. You might go back to the first, but also assume that rockfish and flatfish densities in the drop camera are correlated with rockfish and flatfish densities in the bottom trawl occurring at the same place, but that maybe there's a coefficient for that relationship. There might be this sort of capacity. You say 13 minutes, but I've got 9 minutes more up there. I'm so confused. Okay, I'm going to speed up again. I'll just take the number I like. That's my but okay, so anyway, I mean, you can look through this, and there's different structures that you could think about estimating in this in the relationship between these variables. AIC, like a model comparison metric, thinks that the most parsimonious one is this second most complicated one. And so if we see that and start looking at the coefficients in detail, we see that sponges has a positive and significant effect on rockfish densities. Corals has a negative and significant effect on flatfish densities, or association with flatfish densities. That the coefficients for the additional impact of sponges and corals on rockfish is low and typically non-significant relative to the effect on and then these coefficients are proportional. So the drop camera density estimates are proportional. The variation in the drop camera is proportional to what we see in the bottom trawl. So we think that the drop camera is sort of measuring the same thing as the bottom trawl. Finally, we can use this to predict densities that we would have seen in the bottom trawl, even in places where the drop camera operated, but the bottom trawl did not. So this is a portion of the Aleutian Islands. It's this kind of shallow bank called Bowers Bank that stretches north off the Aleutian Islands that we didn't typically, we have never bottom trawled, but we did do the drop camera survey in. And we can use the drop camera survey to measure flatfish, rockfish, sponges, and corals, and then predict bottom trawl equivalent densities of those fish in an area that we haven't surveyed. So that's what we show here. So I guess the take home here is that there's this growth field of causal statistics for big data. We don't want to. Personally, I don't think that the goal is to replace us all with a big large language model. Instead, the goal is to, think about how we can use our scientific knowledge to make inference, not just predict things, but also make inference about ecological processes. And so these structural models are possible for big multivariate spatial temporal data sets. There's an expressive interface where you can, the user can specify a lot of domain knowledge in a simple way. That I think is easy to teach and fun to explore. Using these causal statistics for big data, we can do global synthesis in this more ecologically informed way. So we can, for instance, get at trait trade-offs that make sense given, ecological theory. And we can also estimate, we can go beyond this sort of species associations to make inference about the mechanisms that give rise to the patterns that we see. Okay, yeah, I don't want any questions anyway, but I will accept them. And yeah, no, that's a joke. I'm sorry. I should clarify. Yeah, so Rob asked a great question about kind of how much phylogenetic signal there is in the trait database in the analysis that I presented. And so the short answer is that I don't know for that data set. So for fish life, we used an Ornstein-Uhlenbeck process, and we didn't test different transformations of the tree.
Right.
Since then, I've sort of worked with Wouter van der Bijl, who's at UBC, to develop a more user-friendly package called PhyloSem. And so PhyloSem allows you to estimate Poggle's lambda and delta and the OU and these sort of this toolbox of transformations of the tree. When I've subsequently looked at things like the trade-off between growth rate and mortality rate and maximum age, it's been a high, the additional variance that's not explained by the tree is very relatively low, like 15%. It would be great to revisit that for a big tree. I just haven't done it with the full data set. Yeah, and Dang also has an amazing question. I couldn't say which of these is a better question. About whether in the future FISHGLOB would be likely to use FishLife or would be likely to use PhyloSem. And so in NOAA, we talk a lot about this, like research models and operation models. And I think that FishLife is a research model. It has a bunch of additional flexibility for nonlinear phylogenetic modeling, like stock recruit modeling. And, it's been sort of growing by accretion over about 10 years. PhyloSem is the really simple to maintain, easy to use, easy to document on CRAN version. And so if there was interest in like FishBase or other partners kind of operationally using and updating phylogenetic trait imputation, I think PhyloSem is the way to go. But I also think you always need to maintain sort of a research model and an operations model, and you can't confuse, you shouldn't confuse them.
So maybe we can go to the next presentation also by you, Jim, in collaboration with Aurore. And if there is additional question about your previous topic, we could take care also.
That's great.
At the end, so when you want to, you can.
Well, thank you again, Bastien. I used all of my jokes in the first talk, but here I am. So this is actually a talk that I wish Arnaud Gruce could have given. Arnaud is a colleague in NIWA in New Zealand, and Aurore Moreau and I have been lucky to work with Arnaud in a couple ways, one of which is this paper. I kind of volunteered to give the talk, but thanks, Aurore, for all the effort involved. And thanks for helping to prepare the slides with me. Yeah, so here's a picture of Arnaud. New Zealand time being difficult to make here, but Henning Winker is at JRC, or I think he's in ISPRA. It doesn't. Okay, FAO, yeah, and then Nicola Walker's at CFAS, and Nathan, I actually don't know where he is now, but thanks to the authors. Yeah, I shouldn't guess, but I should have prepped that ahead of time. But yeah, we published this in Journal of Applied Ecology recently. So we were trying to think about sort of applied and fully worked examples of how something like FISHGLOB could be used for conservation biology. And one application is this IUCN Red List, or the International Union for the Conservation of Nature, is an organization that has been maintaining this Red List assessment. So the goal of the Red List, as I understand it, is to convene panels of experts that go through and score huge batches of species. Either they're not evaluated, they're data deficient, or then they're classified on this continuum of extinction risk, least concern, near threatened, the threatened categories, extinct in the wild or extinct. And so it's an independent process, independent of government entities, independent of national jurisdiction for the public and consumers to understand something about the status of nature. And there's plenty of evidence that, having a management system based on both public opinion and harvest control rules will lead to benefits for conservation. So the IUCN Red List for marine, for marine species, well, the IUCN Red List in general follows these different criteria. So the 5 criteria are declining population size, smaller declining population range, smaller declining population size, very small population quantitative risk assessment. So there's formal criteria for each of these that I won't get into, but the declines for, criteria 1 and 3 are assessed relative to generation time. And so it's helpful that we have this fish life, this sort of phylogenetic trait imputation where we can predict generation time for all fishes. And so the steps that we propose are to fit a spatial model to the data in FISHGLOB, to then extract an estimate of biomass and fit a population model to that biomass time series. We then extract generation time from the life history model, and we compute declines per generation to compare with the red list assessment. So this is sort of a batch process that showcases how we can use FISHGLOB for conservation. So one example is starry skate, Amble Raha Rod. Perfect. In the North Sea. And here we've got an estimated density map where we're trimming out densities that are below a certain threshold. And so we've got it shown here for different selected years from, this 40-year North Sea survey that is one of the many surveys in the FISHGLOB data set. So you can see here that, the distribution used to be relatively extensive. There's been fluctuations and hotspots over time, and that, as density, overall biomass has decreased, there's also been this sort of apparent range contraction. So if we take that density surface that's estimated by the species distribution model and we sum across space, we get this index of relative, index of abundance. We treat as an index of relative abundance. And we get confidence intervals based on the predictive uncertainty from that. And that's what we see here. It's obviously apparent that there's a decline over time, but this is not by itself enough to do the IUCN Red List assessment. The next step is that we pass it through a population model called JABA, which is a type of state space, or, well, it might be, I think it was JARA, actually. It was a state space red list assessment software that Henning Winker has developed. And so here we see it relative to 2020 with the different generation times estimated by fish life shown. And we can get these sort of Bayesian posterior distributions on the annual rate of change over those generation lengths with uncertainty estimates. And so if we line that up with the criteria, we get this substantial decrease over 3 generations, which puts 97% posterior probability in the most critical of the categories assessed. Alternatively, we did this assessment of Atlantic cod that Nicolas at CFAS leads an ICES assessment process for. And so again, we get this, we get an estimate, an index of biomass that we can convert to a biomass index. We pass that through JARA and do these one step ahead predictions. And that's what's shown here for the last 10 retrospective peels. And then we can convert those again to these sort of these different change criteria in any given year. And we see that by the end of this period, there's again, one of these lower, higher risk categories that's identified. So the future steps, this is a pilot study. The idea is to point out that we could use something like FISHGLOB in the sort of international conservation space to scale up across species and across regions. Based on the data availability, there's, hundreds to thousands of stocks that could be assessed this way. And then additionally, the model is producing not just a biomass index, but we have this spatially extensive data set, and we can also estimate the range contraction criteria, and can also use that to compute the IUCN range contraction status. So that would be an interesting topic to explore in the future. I think that's it for Arnaud, and we can take questions. Thanks.
So many thanks, Jim, Aurore, and also Arnaud for this presentation. So, does any have questions?
Yeah. So Malin said, Jim, should I read this off? Yeah. Yeah, so Malin is interested, in the generality of the method, but also curious to hear what species it might not be appropriate for. Yeah, I mean, there's certainly, one concern I've had and worked on is, cases where the stock is only partially observable in a bottom trawl survey. That's an issue with FISHGLOB. We saw that in the previous example with untrawlable habitat, and we have these cases, for instance, where the stock might be changing its distribution between trawlable and untrawlable habitats over time as it rebuilds. So in those cases, fishery scientists typically estimate like a time varied catchability or time varied selectivity, and those are cases where that IUCN fifth category seems, worth the effort. I'd say on the other hand, there's this sort of ecological risk assessment, or HOBDE has the sort of SAFE guidelines, where the idea could be that you try to do assessments for as many species as possible and use that to categorize, prioritize which ones you do like a more quantitative assessment for. And so I think that this is personally, I think this would be a great method to kind of, as a first pass, kind of semi-quantitative method to, prioritize where to put additional research effort. And Malin said, nice. Yeah, let it be known. Well, Aurora, I think, was pointing out that fishes relative to other taxa have fewer species that have been assessed in the red list. Is that right?
Of course, like demersal.
Demersal fishes. Okay, yeah. And I know that some folks at NOAA had as a pandemic project trying to do red list assessments. Monroe, I'm trying to think of his first name, at the Northeast Fishery Science Center, did some flatfish assessments. Again, my hope and my goal would be that we have sort of more data informed assessments for red list and otherwise in the future. I suppose we've got extra time, so I can keep saying that, like also things like management, the Marine Stewardship Council, tries to assess sort of, bycatch risk for other species. And so that's another place where these sort of non-governmental assessments are occurring that could benefit from FISHGLOB, I think. So that I think Rob's question is like, what do you do with the red list assessment in cases where the status is rapidly changing over time? So in that example, I forget which of the two species it was that it went from red up to green back to red. I guess I can check it out here. Yeah, for instance, here where it goes from orange to green back to orange over a fairly short time. And yeah, I mean, I don't want to put words in anyone's mouth, but I do remember Pamela Mace kind of pointing out the difficulty of coming up with criteria that are applicable across all flora and fauna. And there's a trade-off between kind of focusing criteria on individual taxa and then making them too inflexible. And yeah, it's certainly the case that in fisheries management, fishes have these big, some fish life history types have big booms and busts. And, I'd say that this is just information that then would go into some kind of management rule. how you communicate that to the broader public is a real hard challenge.
So you have an additional question from Deng. So what is the minimum level of wearability of the baseline data that is acceptable for this methodology?
Yeah, I thank you, Deng, for that question of So there's really no floor on how bad data can be, so it's always hard to say. I mean, like there's kind of due diligence to make sure that people didn't make up data, and like scientific integrity and these kinds of filters are important. I would say, once the data is from a credible source and has been validated, like we try to do, I typically think that you can try to model it and then make sure that the uses that result is put towards are appropriate for your level of confidence. So I'd say there's a science communication problem again here. Yeah. No, that's a great point. Yeah, there's people, scientists will try to make inference with almost anything. I think it's this would be a great way to replace less credible data poor methods. Another example is that people often use small postage stamp surveys and they assume that that's representative of a population over a broad area. And so the types of spatial analysis we're doing here, if you have only a fraction of the domain of the population that's sampled, they have really high variance. And so the kind of spatial method that we're using discounts information when it doesn't cover the full area. Yeah, repeating that, and I 100% agree that these bottom trawl monitoring surveys are missing some species that we now know from tagging and eDNA, and are also just highly inaccurate for other species that we do see in them.
Marlene, yeah, we have one more question in the room. Yeah.
Yeah, so attempting to rephrase Malin's question, it's given that these methods don't work as well when the survey is only capturing a fraction of the population, how do we figure out which species it might do well or poorly? And I'd say, yeah, it's a topic. It's a good topic for people to be concerned about, and I think that's where you're going with it. And then, I like these sort of spatially explicit models because they provide an avenue for data integration. So, for semi-pelagic species, for Pacific Ocean perch, for instance, we know acoustically that they make these chimneys that go up the water column, and when we do a bottom trawl, we only capture the bottom. And we know that the currents of those chimneys is increasing as the population expands. And so that's a case where we are investigating sort of integrated models that combine acoustic and midwater and bottom trawl. And in that case, we've seen sort of the shift in vertical availability. So I think a lot of these are these limitations are also in invitations for us to think creatively about how to combine bottom trawls with other data types. Yeah.
Is it also an argument for combining across adjacent bottom trawls?
Yeah. So Malin's, is it a good reason to combine across bottom trawls? I agree that, like in the catchability coefficient, availability of a survey is both kind of horizontal availability, vertical availability, gear efficiency, and other factors. And so each of those can be experimentally measured in the field. And so, this type of work is paired well with what we call process research at NIMS, focused experimental studies with alternative gears to, or adaptive surveys beyond the geographic range to try to measure that.
There is another question in the chat.
Let's see, I'm going to try to read it here. I think that the question is, in comparative studies between species, it is common to encounter the need to account for phylogenetic effects. In this context, would using phylosem to impute traits be a good strategy? So I think my reading is that, since Felsenstein in the 70s, we've known a variety of tools for phylogenetic correction in comparative studies, and there's sort of this move to move towards what are called phylogenetic generalized linear models or generalized linear mixed models. And PhyloSem is one example. There's other ones. I like PhyloSem because it's can be scaled up to large phylogenies, like 30,000 fishes. It allows you to have mechanistic structure among traits. But there's still plenty of new work to do, like putting in variation in genetic, evolution rates, different types of transformations for different traits. And so there's a lot of work left to do that I'd love to collaborate with others.
So many thanks for your comments and questions. So now we have the presentation from Laurent Pécuchet, who is associate professor at the Arctic University of Norway.
Hi, everyone, and thanks for coming back after the break. So now I'm going to show a bit of an example of how using FISHGLOB to look at fish community composition. And so this project was to look at spatial and temporal changes in life history strategy composition, and especially looking at how they respond to change in fishing pressure and temperature. Okay. So we have we are looking at this life history strategies that Jim showed also earlier. So it's this opportunistic, periodic, and equilibrium life history strategies, and it was first used by Miller and Rose. So it looked at the trait and their trade-off, especially in the reproductive strategy of fish. And it shows that by looking at the trait and the trade-off, we can basically categorize fish into three life history strategies. So we have periodic strategies, so thinking typically of, for example, cod and Gadidaes. So it's those species that have relatively large lifespan, and have a very high fecundity. Whereas you have, and a very low parental care and very little offspring size. We have the equilibrium species, which have... Oh no, the audio is not on? But it's on here.
Yeah, it's on here.
It's working? Okay. Then the equilibrium species, which have so also long lifespan, but comparatively very few offspring, parental care, large eggs, or even giving birth to youngs. And then we have this opportunic species that is more like, for example, Jim was talking about the gobies. So it's this species that have very fast turnover, and some sign of parental care. And so these three primary life history strategies are also responding to environmental variation, and we're gonna go back to this. So we expect those strategies to be performing better in different environments. So this was a kind of a continuation of studies that are done in 2017, where it was only focusing more on the spatial pattern of life history strategies, and we're at this time only using the European waters and Greenland and Iceland. And we saw that there was quite a fairly strong spatial patterns in the proportion in the community of life history strategies, with, for example, more type of equilibrium species in Northern Europe and towards more polewards, whereas that in the Mediterranean Sea, which was the Spanish Mediterranean Sea, and in the Southern North Sea, for example, there was much more opportunistic species. And these life history strategies also seem to respond to the environment, so especially sea surface temperature and seasonality. And then during the study, we just had taken the North Sea region to look if we could see also changes in time. And when we did this, it was actually quite a strong temporal signal, where we saw that in the North Sea, it seems to be, so across the region, seems to be an increase in equilibrium species and opportunistic species, whereas there's been a decrease in the periodic species. So it made us wonder a bit, like, should we expect that one life history strategies kind of take advantage of the global change more than others strategies, especially thinking more of maybe the fast life species, which are more typically opportunistic species. And there were some other studies that kind of showed this, that this small size, fast life species seems to maybe taking advantage, but there was other studies that actually saw the opposite, but more periodic species appear to do well. So we just wanted to have a look at it. If we see a kind of response. And then second question was, can we try to explain the temporal change so of the life history strategy composition in response to climate change and fishing? So first, we know that there is a spatial heterogeneity in how the environment and anthropogenic pressures have changed so here we look at from 2003 to 2020. So first, if we look at bottom temperature, in that kind of short period, 2003, 2020, we saw that there is some area that have been warming. So this is here in blue, whereas some areas have actually been cooling a bit. And there's been also a lot of variety in this rate of change. The same if we look here at predictability of bottom temperature, we see a lot of spatial variability and also in the changes of demersal fishing pressure. So when we look at that first, we of course make the hypothesis that the environment is changing in a heterogeneous way. So we also think that we're going to see heterogeneous change in life history strategy composition. So we used the FISHGLOB standardized survey data, and we used the public data that is standardized, and we had some agreement to also use 3 more surveys, which was the Iceland, Greenland, and Newfoundland. And we restricted to the time period of 2003 to 2019, so that we could cover all areas. And for each grid cell, we calculated changes in life history strategy. So the life history strategy, so we had around 1,000 fish species that were caught in the bottom trawl surveys of these areas. And for each of the species, we had some trait values. We use a trait database in Pangea from Esther Bokov that she led. And we had values on size, fecundity, offspring size, parental care. And then we use this trait dataset to calculate the life history strategies. And we did this using a type of cluster analysis called archetype analysis. So it means that, so here in this figure, each dot represents one species or one taxa. So each species is a combination then of different strategies. And so here we find again the gobiidae that are typically much more opportunistic species. We have sharks, rays that are more into the equilibrium strategies, and then we have Gattidae and flatfishes, Prornethidae, which are more in the periodic. So I'm going straight now to some results, and then first looking at the region at the ecosystem scale. So ecosystem, I mean basically a survey scale. So here you have different surveys, and here you have the slope of a linear trend, and each color represents a different life history strategy. Red is equilibrium, green is opportunistic, and blue is periodic. So we also did global model across to see there is overall one strategy that seems to increase more, and it seems that periodic overall is a strategy that increase across the whole ecosystem, but it was like the whole study area. But actually, if you also look per ecoregion, it's there's only few single surveys where they are the strategy that has increased the most, but overall, it's been the strategy that increased the most. And then, so we see here also a bit of variability. So there is some surveys where the equilibrium strategy is a strategy that has increased relatively the most. Whereas there is some survey where we have the opportunistic species, which first we thought maybe it was a strategy that would benefit or take advantage from global change. Actually, it seems to have been a strategy that has decreased in many of the ecosystem. Then if we look more spatially, how it looks like. So here, each grid cell is color-coded based on which strategy had the biggest change in abundance. And it's in yellow if it's actually negative change or not, or if it's not in yellow, then it's a positive change. So for example, here in the Southern Barents Sea, it means that the equilibrium species has been the life history strategies that has changed the most and has been an increase. Whereas the opportunistic species, a decrease in opportunistic species have been the biggest change in the Northern Barents Sea. So when we see that, we see there's a large spatial heterogeneity into changes in the life strategy composition. It's not completely random because we can see that there is often kind of a bit more like sub-regional changes, but there is definitely a big variation. So there is spatially heterogeneous changes. So then we wanted to see if this heterogeneous change could be due to, as we see in the beginning, that the environment and the fishing pressure has not changed similarly all across the study area. So the life history strategy, this equilibrium periodic opportunistic framework, also come with this theory that try to explain where or under which environmental condition each life history strategy is occurring more or is prevail more. So the periodic species are expected to prevail in environments that are seasonal, but that are predictable. Whereas the equilibrium species are predicted to favor environments that are kind of more stable through time and space and very predictable. And the opportunistic species are those species that would take more advantages of stochastic, like a bit chaotic conditions. So more will take advantage of environments that become a bit unpredictable and varying. So we looked at this by calculating some variables of using temperature and primary productivity. We first look at the seasonality. So from environment that is quite stable through time to highly seasonal environments. So we expect that, for example, periodic species and opportunistic species will favor a more seasonal environment. Then we have two indicators for the predictability. So contingency is like that from one year to the other year, the conditions are kind of coming at the same time. And in that case, so equilibrium species, we expect they are positively reacting to more predictable environment, whereas opportunistic species will be negatively. And then we also have the color, which is more from a time step 1 to time step 2. Is there some predictability? So in a sense, is there some autocorrelation in time? And this will be positive for periodic and equilibrium, whereas we expect it to be negative for opportunistic. And then we also have some data on fishing pressure. And fishing pressure has been shown in the literature to especially impact these long-lived species that are equilibrium species. So we expect a negative relationship with fishing pressure. So this is just to summarize a bit this hypothesis, but I will just go on. And then we used globe color and Copernicus to calculate these different indicators. And then we also had fishing pressure data from, we took from Frasianal, which is the destructive demersal fishing, and it was the change in time. And so for each grid cells, we have values of changes of fishery strategies, and we associate them with changes in the environment. And then we did this linear mixed effect model using all data sets, but taking into account surveys as random factors and with spatial autocorrelation. So I'm going now to the results, and especially do we see what we expected? So first I must say that this is still kind of ongoing, so it might change a bit, but I think much of the same, it will probably stay very the same big picture, but there is some of our expectation that seems to hold. For example, we saw that aquarium species tends to decrease in area where fishing pressure have increased as expected, whereas we have, it has been the opposite that what we expected, for example, for the periodic species, where they were not more present in areas that were more seasonal or more predictable. And this was, so it's especially the Caribbean species that seems to have responded as we expected. So here it's on the left showing how Caribbean species increased in areas that are more predictable. So here it's areas that are more predictable in time. We see an increase, whereas when fishing effort has been increasing, the proportion has been decreasing. But overall, for now, conclusion for this study is so that we see these heterogeneous spatial patterns, and it's not, I mean, it's what we expected, and we will see in other studies coming after that we see this often. Overall, it seems that it's the periodic species that have, in our study area, that is only Europe and North America, that seems to have increased, and not the opportunistic species. And maybe it's pointed to a recovery of aquarium species with decreasing of fishing effort, but we will have to look a bit more into that. And we see some deviation of the responses of periodic and opportunistic species to change. I listed here some limits of the study, and that, of course, something that we have to especially remember is that we are comparing some very different ecosystem. So we have really this heterogeneity of ecosystem, but different contexts. And so while we look at this big picture, I think what is really interesting also is to also keep this kind of regional views of what is happening. I think I will stop here. Thank you.
Many thanks, Ren. Another question?
Takes a little while.
Yeah.
But maybe we can also take at the end of the session, if we are a bit late already.
Okay, there is a question.
I think depth. So because here what we looked at, so depth would definitely play a role in the spatial pattern. Right now we are focusing more on the temporal changes, and trying to explain temporal changes with changes in temperature and fishing pressure. But it could be interesting to see if we see also some changes, if changes are more prevalent in certain depth, for example, stratum, but we haven't looked at this. Yeah, this one there. Across the grid cell, yeah, across the grid cell for sure. We have some stations that are more like some grid cells that are a bit more coastal and some that are a bit more in offshore area. We did keep only, I think, stations that are more than 20 meters deep. So like try to remove a bit the really coastal one, but there is some depth variability. Yes, good question. I think so we did include in a way pelagic species in this. So we kept all the species that are caught in the bottom trawl surveys. So pelagic species are also, although not how do you say that? Although bottom trawl survey are not made of pelagic species, they are sometimes caught in the bottom trawl survey. So we kept them, but they are maybe not very well standardized in that sense. But yes, it could be that pelagic species have a different response.
Okay, many thanks for your question and comments. And thank you, Lauren, for your very interesting talk. Now we are going ahead with a presentation of Zoe Kitchell, who is postdoctorate at the Occidental College in USA. She's going to talk about species compositions change through time. So you're welcome to start whenever you want.
Okay, thank you so much. And thank you to all of you for still being here. There's so much exciting research and opportunities for collaboration to share, but I also know it's a long time to sit in front of Zoom. My name is Zoe Kitchell, and I'm a postdoctoral researcher at Occidental College in Los Angeles in the States. And today I'm going to be presenting another research application of both the FISHGLOB Consortium and then also the public data set. So the work that I'll be presenting on today talks about testing for changes in spatial beta diversity in marine fish communities through time. And it's integrating the public data sets available through FISHGLOB, but then also other bottom trawl data sets that were, I was able to use them through collaborations and co-authorships with folks who organized those many surveys. And it's no surprise to anyone on this call, but species in the ocean are moving where they live. And these are just three examples that I'm familiar with in the Western Atlantic or the Northeastern United States, American lobster, red hake, and black sea bass. And I won't spend too much time here, but one thing that I immediately noticed by looking at the changes in their distribution through time is that they are not moving in exactly the same way. There's immense variability in how different species are responding to environmental change and also resource extraction in marine environments. And so that led us to use the FISHGLOB Consortium and data to ask the question of what are the impacts of recent species range shifts on regional community composition? One potential outcome is biotic homogenization, where across space, communities of fish start to look more like one another, and therefore they lose their individual uniqueness across space. We may also potentially see the opposite consequence, in which we might see biotic differentiation, where communities of fish end up looking less like one another across space through time. Both homogenization and differentiation can impact ecosystem stability, resilience, and also availability to people reliant on the fishing industry. We think in ecology a lot about homogenization, but less so in marine environments, and also less so in the context of native range shifts. We often think about homogenization in the context of terrestrial or freshwater, non-native or invasive expansions. And so through this research, we were interested in addressing just how does heterogeneity of community composition vary through time. We were able to use 34 bottom trawl surveys spanning from 1968 until 2021 that included over 2,000 fish species and nearly 200,000 individual sampling events through that period of time. And what we did is we calculated trends in dissimilarity or beta diversity spatially through time. And I am showing this graphic here to give you a quick sense of what that actually means. Just for one example region, this is the Eastern Bering Sea and the Northeast Pacific. And each one of the points on this map shows a sampling location. And so what we did was we calculated the dissimilarity between each possible 2 sampling points. And then we divided that by the total number of pairs within a particular survey set. And that gave us a single value of dissimilarity for each year within each survey region. And specifically, I won't talk much about this, but we selected the Jacquard Index of Dissimilarity. And although abundance data is available for many of these surveys, we focused specifically on occurrence data for the primary analyses. To give you a sense of what we found, I want to first talk you through the structure of many of the visualizations that I'm going to use moving forward. On the x-axis, we have time starting in the late 60s and moving until a few years ago. And then on the y-axis, we have Jacquard similarity, annual Jacquard dissimilarity. So if you were to see a pattern decline on this graph, that would represent a region that's experiencing homogenization. And if you see an increase on the graph, that would represent a region experiencing differentiation. So to give a few examples, I'll start with Iceland. This is a survey region where we found that there's really no directional pattern in dissimilarity over time. So this is a region where we did not find evidence of either differentiation or homogenization. In contrast, we also included observations from the west coast of the South Island of New Zealand. And there is a bit of a, maybe like a decadal cyclic variability that we're seeing in this region, but we also found that overall there was a positive slope, and therefore this would be an example of a region that's differentiating through time. And lastly, this is an opposite example of a region where we found evidence of homogenization. And this is the Southeast United States in the West Atlantic. But as I said before, we included 34 survey regions. So they are all included on this plot here. And if immediately you're not able to pull out any specific signal, that is exactly what we found. So although for many regions we found significant trends either towards differentiation or homogenization, the overall trend was that there's no general pattern of either homogenization or differentiation found across trawl surveys in really diverse marine ecosystems worldwide. And here's a map of how those trends varied across regions. The areas that are a orange or yellow color, those are homogenizing. I'll highlight just a few right here. The regions that are in like a pink-red color are those that are differentiating, and then those in a blue color are those that are showing no significant change. So we did find evidence of homogenization in 10 out of the 34 regions, differentiation in 11 out of the 34 regions, but overall, there were more regions not changing or not showing a directional change than those that were exhibiting a directional change. So what we found is that spatial dissimilarity is often nonlinear through time, and it varies by region. And so then we were interested in looking into some of the potential drivers of changes in dissimilarity through time. And specifically, we looked at regional sea bottom temperature, and we used a number of different metrics that I won't go into now. And then we also looked at regional fishing pressure in the year previous to when the survey occurred. And again, what we found was that there was a lot of variability across these different regions. So we found no overall relationship with temperature, but strong regional patterns. And so I'll zoom in on just a few of these. Looking at the Rockall Plateau in the North Atlantic, what we found was that cooler years were more homogenized and warmer years were more differentiated. But then if you hop over to the Baltic Sea, you find the opposite pattern, where warmer years were more homogenized and then cooler years were more differentiated. And now we'll pop over. It's interesting to note that all three of these regions are actually quite close to each other, but exhibiting really different patterns. So this is the shelf off the coast of Scotland, and here we found really no strong relationship between dissimilarity and temperature. And so we asked similar questions in relation to fishing pressure. And again, no strong overall relationship across all of our regions, but lots of strong relationships within individual surveys. So we're going to start in the North Pacific in the Eastern Bering Sea, where in years of higher than normal fishing pressure, you saw a more homogenized state, while in years of relatively low fishing pressure, you saw a more differentiated community. And if you jump over to the Atlantic side of the United States, we again saw the opposite trend. So in this case, when you had years of relatively high fishing pressure, those were associated with more differentiated years, while years of lower fishing pressure were related to years of a more homogenized overall bottom fish community. And again, of course, we had some regions that really didn't show any strong signal between fishing pressure and dissimilarity. One example would be the east coast of the South Island of New Zealand. So to wrap up, a few conclusions from these analyses that allowed us to include, due to fantastic collaborators and data providers worldwide, allowed us to look at a lot of really unique marine ecosystems experiencing very different types of pressures and environmental change. What we found were dynamic patterns of homogenization and differentiation over time. We found that neither homogenization or differentiation were more common than no trend through time. And our biggest takeaway was that the impacts of temperature and fishing pressure were super highly context dependent. However, it's important to acknowledge that some regions were homogenizing and ask critical questions about why that might be in those particular regions. I'd be really excited to see future researchers, potentially some of you, dive into like a single region and ask maybe like, why are we seeing homogenization in that region? Because we do know that homogenization has the potential to lead to instability of the ecosystem and perhaps a decline in resilience. And I also just wanted to highlight that the more differentiated fish communities have the potential to help sustain regional fisheries in light of environmental change. So I will end there, and I think I have time for some questions.
Many thanks, Doris. So there is questions about scale.
Fantastic. Thank you so much, Shahar, for your question. How did we choose the size of the region? We thought really critically about what scale was most appropriate for this study. And in some ways, we chose to not make that decision because with this type of temporal diversity analysis, we weren't at the point with the data set where we felt comfortable like fully homogenizing all of the surveys. That would require some higher level modeling that we look forward to doing in the future, but haven't done yet with our homogenized data set. And so what we did was we just used the footprint of each of the surveys and the way that they're collected. So as you probably noticed on the map earlier, there's some surveys that are quite a bit smaller than other surveys. We did check whether something like survey size had a big impact on the trend that we found, and we didn't find any clear relationships between survey footprint size and the trend that we detected. But we did not decide to kind of like standardize all the survey sizes because we wanted to be able to integrate as much data as we could. But I know from other work looking at trends in spatial beta diversity over time, that there do tend to be differences if you're looking at like maybe a small stream or a pond versus if you're looking at like a full like landscape, or if you're looking at a country. You do often see different likelihoods of homogenization and differentiation, especially because the number of species living in a habitat often scales with size. But we did not find a strong signal in our work. But awesome question. Thank you for asking. There's another question. Do we think that we would have the same results with abundance versus occurrence? Thank you so much for asking that. I focused on the results from the occurrence observations today, but we did also look at abundance data in addition to relative abundance data. And the general takeaway that more regions exhibit no trend in spatial beta diversity compared to either homogenization or differentiation is the same. But was interesting is that there were some regions that would switch their classification depending on whether we were looking at abundance data or occurrence data, which I think definitely warrants more research and more exploration to get a better sense of why that happened for particular regions. But for the most part, the overall takeaway of this work was consistent no matter which metric we are using. Okay, we have a question from Cher Chow. If the Jacquard index is calculated by comparing between time points and not locations, okay, I think I'm understanding this correctly. So what we did was that we calculated Jacquard index between sample locations, and we did not calculate Jacquard index across time. It's a little confusing because you can look at spatial beta diversity over time, but then you can also look at beta diversity between time points, and that is not what we chose to do in the study. But I think that what's fantastic about this data set is that's absolutely a question that could be asked about this data set as well. And I know folks have specifically looked at that question with some of our surveys, including the Scotland Shelf Sea. And they have identified trends in temporal beta diversity as well, but that's not what we looked at here. And I just want to check in on I'm happy to answer questions over the chat once I hit my time.
Great.
I have time? I'm at time. Okay, so I see a few other questions, which I look forward to answering, but I'm going to answer those in the chat so that we can move on to our next speaker.
Okay, many thanks, Erin. So now we are moving to the next presentation about marine heatwave and change in biomass and composition of marine fish. So a presentation given by Alessa Preston and Hélène Soubelet, who are researcher at the University of California, Santa Cruz in the USA. So please.
Great. Thanks so much, Bastien, and thanks so much to everyone who's stuck with us online. It's been a wonderful set of talks from my colleagues here. So I'm Malin Pinsky. I'm faculty and director of the Global Change Research Group at the University of California, Santa Cruz, and I'm joined by Dr. Alexa Fredston. It seems like I can't tell the video. Yeah.
And the simulator.
Faculty member in Ocean Sciences, also at the University of California, Santa Cruz. So this figure, or something quite similar to it, is something you're probably quite familiar with. Here we've got the increases in the land surface temperature in red and in the ocean surface in blue. And this long-term warming has been a topic of research for many different groups around the world. Lots of interesting research going on. But warming also happens very quickly during relatively short but extreme high temperature events. In the ocean, these are often called marine heat waves, and they're shown on this map here. They can last from a few days to a few months, and they can have very large impacts on plants and on animals. And in fact, there are good reasons to expect that these extremes have even larger impacts than the long-term slower warming trends from the previous graph that I just showed. One of the strengths of observations like FISHGLOB, because it covers so many ecosystems and a wide geography, is it allows us to test hypotheses and understand how general processes and responses are across many different ecological and environmental contexts. And at this point, the scientific literature is quite full of actually quite dramatic reports of large marine heat wave impacts. Everything from die-offs of kelp forests to entanglements of whales to reproductive failures in seabirds. So for the analysis you're about to see, we used the public surveys on marine fishes in the FISHGLOB dataset. These cover much of North America and Europe. And these surveys cover a time period and locations of many marine heat waves. One of those examples is shown here in the upper left. You can see the spike that happened after 2015. That was a marine heat wave in the Gulf of Alaska. It had a cumulative intensity of more than 40 degree days. And intensity here is measured as the cumulative degree days over a seasonally varying 95th percentile of historical sea bottom temperatures. So in total, we had data covering 248 different marine heat waves. And we use those for examining the potential impacts of those heat waves on marine fishes around these two continents. So now I'll pass it off to my colleague, Alexa, to talk about what we found.
Thanks, Malin, and thanks everyone for being here. If people have more questions about how we quantified and measured marine heat waves, please put them in the chat, and I'm happy to answer them. But I'm going to skip directly to our results, and there are kind of two major response variables that we looked at in the study, and the first one was just overall biomass in these FISHGLOB surveys. So using the public data sets, how did the whole assemblage respond in terms of its biomass? That's what's on the y-axis, and if it's a positive value, that just means there were more fish in that survey than in the previous one done in the same region, maybe 1 year before or 2 years before. And on the x-axis, as Malin explained to you, is the intensity of the heat wave. These results that I'll show you are very robust to both which marine heat wave metric we used and which kind of biomass or abundance metric we used. And the first thing that jumps out when you kind of see part of the data, we've hidden the rest of it from you, but you'll see it soon, is that there's an enormous amount of variability in bottom trawl surveys, even in years when there were no heat waves. So that's this big cloud on the left. So you can see from year to year, they fluctuate enormously in biomass, and that probably reflects a combination of true biological variability and the imperfect observation process of the surveys themselves. And here you also see some of the what we'll call famous marine heat waves. Malin mentioned the Gulf of Alaska, where there was an enormous heat wave that people called the blob that lasted for years from 2014 to 2016 in the Northeast Pacific in general. And so that was really felt in the survey done in 2017 here, where you see a decline in biomass and a very intense heat wave. The y-axis units here are log ratio, which can be a little hard to interpret, but recognize that, by the time you get into these units of 1 or -1, you're talking about almost a doubling or a huge reduction in the amount of biomass. So the unit might look small, but the differences are huge. Here, though, is the full response in the FISHGLOB data that we compared to marine heat waves. So this is 1993 to 2019. You see, like I said before, some ecosystems that have a major decline in biomass after a marine heat wave, but you also see just as many that have a major increase in biomass. This is once again consistent with what we know from literature. So for example, this Northwest Atlantic marine heat wave in 2012, which is what you see in both the Gulf of St. Lawrence and the Northeast USA, was associated with one of the best ever years for fisheries in those regions. So taken together, there is no coherent global or fish global response to marine heat waves. One question that we asked is, well, maybe some systems have higher biomass after heat waves, others have less, but overall, maybe they just have more variability. And the answer to that is also no. So now we're looking on the y-axis at the absolute log ratio, asking, is it just higher as marine heat waves become more intense, or when they occur versus when they don't? And the answer to both of those things is no. The same heat waves are highlighted, so you can sort of look at your favorite heat wave as an example. The second response variable that we looked at was, okay, maybe biomass is approximately not changing over time, but the community is changing. We looked at that a few different ways. Here we're looking on the y-axis at something called the community temperature index, and this measures the overall temperature preference of the fish. Do they generally like warm water? In which case the value is high. Do they generally like cold water? And what you're looking at on the y-axis is actually the change. So if it increases, that means you had community turnover favoring warm water species. And I'm going to show you the 4 surveys we have that encompass that huge blob heat wave in the Northeast Pacific from 2014 to 2016. And let's start by talking about the Gulf of Alaska, because that's exactly the story we thought we'd find. It's this yellow line, and over the course of the heat wave, which is shown in this gray bar, you just have this huge increase in the community temperature index, which demonstrates a really strong community turnover towards warm affiliated species. But that didn't happen anywhere else. One region had actually a transition towards cold affiliated species, that's British Columbia, and then a bounce back. And the West Coast had a very relatively small change in community temperature index. And the Eastern Bering Sea actually did have this transition towards a warm affiliated community, but immediately reverted back. So completely disparate responses, even within a relatively small geography, to the same oceanographic event. The last set of results I'll present to you delves even deeper into this question of which species did well and which did poorly, to look at the species level. So now we still have change in biomass on the y-axis, like in the first set of plots. But on the x-axis, we're asking, was it a warm water species or a cold water species? And we thought when we plotted these things, we would find a positive slope. So on the bottom left, you would see that species that were relatively colder than the others in that region would do really badly in a marine heat wave, so they decline. And we thought maybe species that are relatively warm affiliated in that community would do really well. Instead, we just found a cloud of points. There's no statistical signal here. So every one of these dots represents one species after a heat wave. Every single one of these was a heat wave, and yet you are just as likely to go up or down in biomass regardless of the temperature preference of the species. Okay, this is last result, sorry. And I'll tell you, I'm happy to answer any more questions about the methods, but just very briefly, we used some extensive power analyses to demonstrate that with the science of this data set, if biomass had declined at least 9% as a result of a heat wave, we would have been able to detect it. We also did really extensive robustness checks and tests for other predictors. So if there's something on this screen you have a question about, please put it in the chat and we're happy to answer that. Back to Malin.
So as you can imagine, we went into this research thinking that we would see these really interesting and clear impacts of marine heat waves, and yet that's not what we found. So lots of additional research ongoing to understand which impacts of marine heat waves or which kinds of marine heat waves are most impactful. But at the moment, there are some potential explanations that can help explain the results that we're seeing so far. One possibility is that it's not just about temperature, that it's also about these compound events, high temperature plus low oxygen or low productivity. Another possibility is that over the timescales at which we're observing these particular ecosystems, fish actually have quite a bit of resilience through mobility, moving potentially deeper over the course of days or months, but then coming back and coming out of climate refugia such that the long-term sort of annual scale responses actually are quite minimal. And related to what Alexa just presented about a couple minutes ago, it's possible these impacts are meaningful, but relatively small, sort of less than the 9% change in biomass that these particular data sets would have been sufficient for detecting. So, at the moment, what we're left for these continental shelf ecosystems that are so important for supporting some of the most productive fisheries around the world, that these short-term extreme events from days to months actually, while they can be impactful, and we have good examples of that, marine fishes actually appear to be quite resilient in general. And there's still ongoing, really interesting research to be done to understand what are the limits to this marine heat wave resilience. So for that, stay tuned. And I think we do still have time for questions. If you have them, please put them in the chat, and Alexa and I would be happy to answer them. One question at the bottom. No. A second to bottom. Okay.
How to estimate. Do you want me to take this one?
Yeah.
Okay.
Yeah.
Thank you for the question, which just to, oh, sorry.
No, that's not really lots.
Yeah.
We've got it here.
Bastien's scrolling. Okay. That one. Perfect. Sorry to those on Zoom. We have like 11 screens in the room that we're actually in, but now they're all in the same place, which gives me a chance to answer Julia's great question about how you estimate thermal preference. So what we would love to do in estimating thermal preference is take each species of fish, like slowly cook it in a lab, and then establish its actual critical thermal maxima and minima. This study had about 1750 species, so we don't have those data. And instead what we use are what people call realized thermal niches. So this is a database that we did not collate, but we merged with the FISHGLOB database. Mike Burrows and a large number of colleagues published this database in their research on community temperature indices. So I'll put the database in the chat after this in case anyone wants to look at it. But it uses actually species modeled habitat to extract the temperatures at which they're usually found and then establish some kind of limits to those temperatures. So we use the median value for that, which kind of represents a central preference in theory for the species realized distribution. It's not a perfect way to do this, but it does let us just get a sense of which ones hang out in warmer water and which ones hang out in cooler water, which was what we were trying to get at in this analysis of looking at potential turnover.
Oh. We've got a question from Mike Burrows, too.
Oh, my goodness.
Yeah. Related. But...
Hi, Mike. Well, he said hi, Malin, so maybe it's for you. But
and Alexa.
Okay. Do you think the size of a species' thermal range might influence the response to heat waves? Thermal species might be more sensitive. That's not something we looked at. We did look at some traits, but we did not look at the breadth of their thermal niche. Mike, because we use the median only in this, we really would have missed how broad those thermal niches are, and that would have been a cool thing to test. My guess is that some of these species probably, are so broad that you wouldn't get that much insight there, but others wouldn't. So I think it's a cool idea.
Yeah.
Do you want to chime in?
There's another question.
Maybe just pointing out, and based on some of the interesting work, Mike, that you've led, I think there are some good reasons to think that might be important, and it would be a good hypothesis to test with these kinds of data sets. That would be a lot of fun to do. And one of the wonderful things is that we do have the data available because of these FISHGLOB data sets being public and now harmonized. These kinds of questions become much easier to ask in a way that we wouldn't have been able to ask before. Yeah. Okay. So got one question. It says, great study from Melissa K. I find it super interesting that the Southeast USA had different responses to 1997 versus 1999 heat waves. Any thoughts on what may be driving the different response there?
Good one.
Yeah.
Hi, thank you for the great question. So often when we see really different responses close in time, some of it involves some reversion to the mean. So one of them was just a really extreme change in biomass, and then the time series is kind of regressing approximately to where it was. The Southeast US survey is a relatively small survey, and it has enormous year-over-year variation in overall biomass. Some of that is I think the ecosystem, and some of it is probably the survey. The only significant statistic in this whole paper was that the more samples a survey has, the less biomass variability you find over time. And the Southeast US is one. I'm actually looking at the time series right now, which is an extended data figure 10 in the paper, if you're interested. But it just kind of goes like this during that time series. So how much those big biomass changes are related to the marine heat wave versus related to another ecosystem process versus the survey variability is kind of beyond our capacity to disentangle. But I think the exact type of regional question that we hope to move on to from these studies, because we don't really know. Do you think that's fair?
Yeah, and I guess I'd also say, this is, one of the, I think it also illustrates one of the ways in which it's also really helpful to maintain and strong lines of communication and also hear from experts within each region. We don't know every region in extreme detail. So hearing from scientists working on the ground in the Southeast US do have experience and hypotheses that can be tested across these different regions would be a wonderful next step as well. We also have a question about time lags. Do you want to take that one?
Sure.
Okay.
And I think we'll maybe say this is our last question, but thank you. So this is a great question. And the question is did we explore a lag in response? We did explore lagged responses of marine heat waves that happened up to 5 years prior to a survey because of exactly the theory that you've put in the chat, which is that maybe there is just sort of a recruitment effect and it takes a while for that cohort to age into a survey. There wasn't a significant effect of even, marine heat waves at any time lag on any of the survey biomass. But it's worth pointing out that one thing we didn't do, which again, to Malin's point, would be a way to take advantage of more in-depth knowledge about regions and species, was connect the biology of an individual species to the time lag that we use for it. So we kind of use the same lags for every species. We didn't say, because there were so many species in this analysis, this is how many years post-recruitment we want to look for a marine heat wave effect on species A. So we might potentially, if we had the species-specific trait data to do that, be able to repeat that with more depth. But looking broadly across ecosystems, we didn't find a lag effect. And if there are more questions, put them in the chat and we'll answer them. But yeah, thank you so much for your thoughts and questions.
Thank you very much, Alessa and Madeline. So now we're moving on the last presentation, the last but not the least. It is about the global transboundary pattern and whether applied science for fishery management by Juliano Palacios Abrantes from the University of British Columbia in Canada, and Nancy Schachter from the Bedford Institute of Oceanography, also in Canada.
Hi, everyone. We're still here. We just happened, both me and Nancy, who's right here, to be in Canada, but it's not related to the topic at all. So I will give you a brief introduction about transboundary stocks and what are we talking about. And then I'll be fast because actually Nancy is the one that has all the juicy results to show. So let's go. For those who don't know, to talk about transboundary stocks, we have to go back a bit in time to the 70s, 80s, when the UN decided to finally accept the concept of exclusive economic zones, those 200 nautical miles that each country that has a ocean border can claim as theirs. Obviously, there are conflicts there if you have a country in front of you that is not in that distance, but overall, those were the limits. Within these 200 nautical miles, countries are both responsible and have the right to exploit their fisheries, and they are not obliged to collaborate with each other, but they are highly advised to. And this is because when we draw these invisible lines in the water, we didn't really consider fish distributions. So what ended up happening is that a lot of fish stocks that were just happily swimming around suddenly found themselves crossing these international borders. And so that's what we know today as fish stocks. In here, we're showing the four main categories from left to right. The red one, transboundary stocks, are those who are shared between neighboring exclusive economic zones. The blue ones, straddling stocks, are those who are mostly inside the neighboring exclusive economic zones, but sometimes they go out to the high seas. And then within those straddlings, you have this brown large ones at the bottom that are like that we know as straddling highly migratory. So these are mostly going to be your tunas, your billfishes that like to really swim far, far, far away. And then the discrete high seas, the green ones, who are mostly stocks that are just inside the high seas. Yeah. So for this presentation, we're only going to talk about transboundary stocks. So again, those that are shared between exclusive economic zones. And yes, those are Dr. Seuss fishes. I really like them, etc. So why do we care about transboundary species? These happen to be quite important for both revenue and catch around the world. So this map shows on the land polygons the number of transboundary species, and in the EEZ polygon, you can see the revenue in millions of dollars, the average revenue per year from those species in terms of fisheries. And so some cases like Peru, for example, you have a few, we identified a few transboundary species, but even these few amount of species represent a large amount of revenue for the country in terms of fisheries. You have some cases which might be the opposite. I don't know if you can see very well, but like, for example, Portugal, because of its geographical position, has a large number of transboundary species, but maybe the revenue from these species from fisheries is not that high. As climate change changes stocks distributions, the management of these fisheries becomes complicated. Climate change is, as we know, is changing temperature, oxygen, all these different variables in the oceans. And those species that cannot necessarily adapt in terms of physiological adapt or composition of ecosystems, they move. They will try to find the characteristics or the environmental preferences that they're used to it. And that will complicate the management of sharing stocks. You can imagine, for example, in this the graph on the right, you can imagine a stock that today is discrete in one EZ, and then it moves to the middle and then moves to the other EZ. Or you can imagine a contraction of a stock that is currently transboundary and that eventually becomes discrete, or the opposite. And what it's important we can see in the bottom is that where depends where you are in the EZ, where you are the one who is receiving or given, your impression and the actual size of the stock is going to change, and that's going to matter for management. So here's where I stop, and I'm actually going to pass it to Nancy, who's going to give you a better example of exactly this type of dynamics. Nancy?
Okay, everyone. I'm officially the last speaker, and I'm the last person to thank those who have stuck around. And I'm going to be I'm going to try to end this very on a hopeful note, because it would really sort of cap off all the great science we saw this afternoon. So what you have in front of you is a schematic to show that the transboundary fish stocks, which Giuliano just showed you, all of these global scale problems or global scale patterns, we use those at a regional level to sort of set the guidelines as to how we look at science. We know that the global scale patterns really mean that it's going to be a challenge for unilateral management, but the management alone has to depend on science, and that would be transboundary science. So I'm going to walk you through an example of looking at my region, which is the Northwest Atlantic. But first, to tell you that the first step in sort of the setting up the science infrastructure is to get these two countries together, and here we have Canada and the US, to form some sort of collaboration framework. So we met with NIMS, representing the US, and DFO, representing Canada, and we had a great workshop which led to a whole series of questions that we wanted to be able, and priorities, to set terms of reference so that we could almost foster collaboration to track, understand, and respond to climate variability. There are 3 regions, the Arctic, the Pacific, and the Atlantic, which I will focus on now. So when we go to the Atlantic, here we've got the Hague line, which I drew in, so it's not that great, but it's approximate. Canada and the USA. And the first thing to note that this is a temperature gradient. So you can really see that on the US side, as those ambient temperatures warm, that animals, the expectation is that marine life will generally shift northeast. And here I'm going to just now boil down right to that nautical border, the Hague line, and talk about Atlantic halibut, which is a good illustration of a transboundary dilemma because it is not a fish that is on a list where both countries have a very formalized and high level process to examine and set total allowable catch. It's done a little less formally. So the Atlantic halibut on the US side was actually designated as overfished in 2012. Its stock is very low and it never really recovered from overfishing in the 1850s. The estimated biomass is around 3% of the target, and the rebuilding plan allows only limited harvest. On the Canadian side, we see that it is certified as sustainable, and abundance indices have been increasing since 2005. So what that leads to is very, very different perspectives of 2 different regions that assess their Atlantic halibut separately. It could be any species at this stage, but each of those assessments are done on different management bodies, different assessment bodies on different surveys. And in this particular instance, the US is worried that our aggressive rate under MS stewardship certified fishery, that we might be impeding any USA recovery. From the Canadian perspective, we're sort of like, we have so many, we're not sure why you don't. We think it might be temperature. So this is sort of where we have to learn and taking down those guiding principles that we learned about earlier and figure out how to measure and detect shifts. And this is a table from a paper by Link et al. 2021, pointing out the importance of looking at a suite of shift indicators, including the centroidal biomass, the distance from the border, area occupied, etc. And the point being here is you use that suite to estimate whether the animal has shifted in or out, whether it's expanding or contracting. So the first step to do that, even to measure whether something's going across the border, means you have to integrate those fisheries surveys. So in our region, a group of people at the Gulf of Maine Research Institute, along with Jim Thorson and a couple of people at Fisheries and Oceans, put together a modeling framework for the integration of fishery surveys. And the idea is you can use it on any species. A GitHub repository has been built, also some visualization, and it's being revised as we speak. But the point is we now have that modeling framework, which is almost like a piece of science infrastructure to evaluate each species. But in this area, we really have to acknowledge climate change. It's very close to home. In this area, we're looking at a time series since 1990. We can see that roughly around 2005, the area warming has really accelerated across the whole study domain. So for our analysis, we looked at before warming and during warming, expecting different responses. So here we've got the output from that integrated model. And if you look at before warming, you can see the orange parts, which are the higher densities. You can see that there is a lot of connection and that those are enhanced during the warming period. So now we want to look at shift indicators, trends in abundance in before warming and during warming. And so on the left-hand side in gold, you can see the time series of abundance over time. And you can see that it starts rapidly rising around 2005. And in the US, it's very low, but it also starts rising. And then when we look at the rates of change, we can see that the rates of change within the before warming period were not significant within Canada, but really started to increase in the warming period. And in the US, they have always been slowly increasing. That would be one shift indicator. I'm only going to show a couple so that we can close out the session. And the second shift indicator is the effective area occupied. So here we're looking at the effective area occupied, or the area that's occupied by the animal if you control for the average density. And you can see it's fairly constant, or there are some dips in Canada, but in the US, it really was declining up until 2005. So when we look over at the rates of change, we can see that in Canada, in the before warming period, that the effective area, the rate of change with the effective area was negative, yet positive since 2005 or during the warming period. And similarly in the US, it had been rapidly declining, as you can see here, but then has now increased. So now we'll just imagine that we did all of those shift indicators and we come out with this schematic. And what we found was that the range edge, so the north edge has increased, as well as a bit of the southern edge, as has the center of gravity in latitude and center of gravity in longitude, not so much change. In the area occupied, it has increased and the abundance has increased. So now to tie it all back to the science that's Alps and the distance to the Hague line, they're going this way. The centers of gravity are moving away from the border. So tie it all back to how this would fit into sort of a framework, from a regulatory agency, is we would have sort of package science advice that would be a little bit more detailed than this, but to describe that this is an expanding stock during the warming period, but then we would also include other information that we know, which is that when we project out to 2050, it is expected to be unfavorable for halibut in this area. So tying it all the way back, we have the beginnings of setting up the sort of science, bilateral transboundary science, and it can only grow from here because climate change is not going away. This will continue to be a problem and quite necessary to do. And so in closing, first of all, I think I speak on behalf of all of us. We're really absolutely thrilled that you all attended, and we're sorry we couldn't be there in person, or that we wouldn't be able to drink cocktails with you. But no, seriously, on behalf of all of us, it has been really great. All the questions are really great. And if you have any interest in FISHGLOB, here's that QR code again. And we want to thank all the contributors who might be online as well. And that's it. That's the end.
Nancy and Juliana, so yes, if you have any comment or question, please.
I'm going to repeat the question. The question was, do we compare the US halibut assessment and the Canadian halibut assessment in any discussions? And is there any genetic evidence that they are separate stocks? So I'll answer the first one first. There is no evidence that there is a genetic difference across the domain of study that we looked at, but there is spatial structure. So there is evidence that on the US side, those animals stay there more often, and some come into Canadian waters. On the Canadian side, those creatures stay there more often, and sometimes they spill over to the US. So there is exchange and enough to swamp any genetic variability. And the first question is, it's a little haphazard right now. But as the value of halibut goes up, I'm sure that it'll become more formalized. What currently happens right now is that a group of us will get a call from the Canadian government, sort of a different international fisheries department, who will just ask us what's the latest science, and then similarly that's happening in the US, and then they meet in Boston. We have an official, more formal process called TRAC, Transboundary Resource Advisory Committee, but it's only for 3 species. So this is something that we are going to have to really consider expanding and formalizing and setting up the governance. Yes, I do. No, in all seriousness, I think that the transboundary science efforts, it just provides enough evidence for these the cooperation on management to be that much easier. So right now, in the absence of any scientific evidence, it will be going in and just fighting for more total allowable catch. But in this way, there's given the whole body of evidence, I just showed you one part of it, there would be sort of a procedure that we can go about estimating total allowable catch along that border. Okay, I'm going to read this wonderful. Many thanks for FISHGLOB and all the conferences, was amazing. Hoping to see more studies related to freshwater fish that are very infected.
Also very transboundary.
Yeah, also very transboundary.
Very.
Do you want to?
So many thanks, everyone, for having attending this conference. We are very pleased to have you during these last hours. Just to thanks again our support, so the FRB, CESAB, CI, the French Embassy in Canada, and the University of Montpellier. Just also to say that we have recorded this conference, so we will make it available soon online. We could share the link. It should be on the YouTube channel of FRB. And so we will provide the link soon in order if you want to share it or maybe have a look again of some of the presentation. Thank you so much again, and hope you will follow our activity. And if you wish to join us, again, you can join us through this email and also the online form of on our website. Thank you.